Good morning, Manala. Professor Anita Mahadevan has been neurophysiologist at the National Institute of Mental Health and Neuroscience, Bangalore, India. For the past 20 years, where she is actively involved in diagnosis, research, and teaching in the field of neuropathology. Professor Anita served as the coordinator for the only human brain tissue repository in the country which procure and provide characterized human brain tissue and for scientific engage in the brain research. Additionally, she spread neuroscience education for school students and promotes brain donate awareness among the public. This faculty has been instrumental in evaluating the role of mitochondrial dysfunction in neurotrauma and contributing to the human program in the field of neuroinfection. Her lab has concentrated on studying the cellular reservoir in the brain of HIV, investigation, radiopathology, correlation of neuroinfection, molecularly characterized HIV subtype in the brain, and exploring HIV-related neuropathosis. Additionally, she has made significant contribution in understanding the pathobiology of ribs, viral encapsulates in the realm of epilepsy. Her lab has played a pivotal role in elucidating the pathology of status epilepsy and developing the RAD module for the hot water epilepsy. Anita Mahadevan international experience include various programs, contribute and participate in different countries for diverse field of study and research institutes, reflect in the wide range of publication in various national and international journals. Currently, her research is focused on evaluating glial biology in epilepsy and central nervous system infection. It is a great privilege to have you here to this conference of speaking.
Thank you very much for that very kind introduction. First of all, a very good morning to everyone here. Am I visible? So, first of all, I think thanks to Dr. Rao for his very kind invitation to be here. Is that okay? Thank you, Dr. Rao. It's a great pleasure to be here in front of all of you and to share a little bit of the work that we have done. It's just a hope that this will be an inspiration to all of you to take it further. Okay? So what I'm going to be talking to you today is a little bit about a brain bank. Now, how many of you have heard of a brain bank? Hands up. Other than Dr. Jagannath Rao. No one? How many of you have heard of banks? Everyone. Very good. So this is going to be a little different. Okay? There is no money in this bank, but it is something that gives you a lot of hope for the future. Okay, so let's try and find out what this is about. Can I have the next slide? Thank you. So we'll start with answering a few of these questions. The first is, what came to all your minds? What is a brain bank? Okay, why should we have them? How is it going to help you? Which is the first brain bank in India? How did it start? And all of the difficulties and challenges that we faced in the last 3 decades. Okay? So this is what I'm going to cover. You are free at any point of time to raise your hand and ask me to repeat if there's something that is not clear. Or if you also want to give your input, I'm very happy with it. You can also ask me to stop. If you want me to stop, stand up. Alright? Okay, so we'll start with the first question. Can I have the next slide? I think you're all very familiar that there are so many brain disorders, right? I can fill many slides with the type of brain disorders. But the fact is, everybody throughout the world is trying to find the best cure for several of these. Be it dementia, be it Alzheimer's disease, be it multiple sclerosis, so many types of brain diseases which unfortunately don't have a cure. Can I have the next? Yeah, so the goal that everybody's after is to find a cure. To find a cure, we need to have a lot of ammunition. It's like war, right? We're all looking at war on television. Even in real life, we are at war at all of these diseases because you are trying to find a cure. Can I have the next slide? So, to help us, what is the kind of ammunition we have to fight? We have lots of ammunition. We have everything from now molecular biology. We have so many omics. Can you name a few omics? The only one I know is comics, but I think all of you know a lot of omics. Come on, come with me. You can make up names. If you're looking at protein, it is proteomics. If you're looking at genes, it is genomics. If you're looking at metabolites, it is metabolomics. Exactly. So you have a whole lot of omics. You also have a lot of biotechnology tools, which is what I think Dr. Rao is just telling us that, for anything to be successful now, you need medicine and engineering, technology, everyone to come together. And your institute is one such which has everything, all under one roof. So if discoveries have to happen, this is the place because all of you can talk together. So, and we also have a lot of animal models. Okay? The next speaker will be talking to you about C. elegans. So we have a lot of animal models too. Can I have the next? Okay, thanks. Now, all of you are familiar with the most common animal model, that is the poor lab mouse. Okay? I found this very interesting thing on the internet which said, see the curriculum writing of a lab mouse. Very, very impressive. Look at the number of Nobel Prizes it has contributed to. Right? But problem is, despite all of these studies, all of this discoveries using a lab mouse, it's not good enough because we still don't have a cure for several neurodegenerative diseases. Okay, so it is a lifesaver, but obviously not good enough. And why is that? Next slide. For one simple reason. A mouse, how much do you think it weighs, a mouse brain? Come on, is it in grams or is it in kgs? Grams. Yeah, right, it's a few grams. Whereas, how much does a human brain weigh? It's 1.5 kgs. So you are trying to compare a few grams to 1.5 kgs. The reason why we have such a big brain, it's supposed to be the most evolved, supposed to be, simply because we increase with the number of neurons. So if your nerve cells in a mouse is just 5 million, you have something like 86 billion nerve cells in our human brain. Which also means it is one of the most complex because of so much wiring. An engineer would understand that it is very, very complex wiring. Okay? So you are trying to compare this with that. Is it possible? Raise your hand. How many of you think that it is easy to compare? Nobody? Very good. Can I have the next one? So, because of which we know our human brain is most evolved. Can I go to the next one? So the question is, we are still using animal models, but which is the animal which is probably closest? You would ask, why can't we use chimpanzees? Right? Many of us are like monkeys only. So chimpanzees you would think is the closest. Now interestingly, if you look at humans, if you look at chimpanzees, gorilla, orangutan, all of these, we share the genes that regulate our survival. We share several genes for DNA repair, oxygen chemistry, blah, blah, blah. So we do share a lot of genes. But although if you look at similarity, it is like 99.4% similar. We are still not chimpanzees, right? Not changed. It also means that we really can't use this. The most obvious is that chimpanzees don't develop several diseases that humans do. For example, AIDS, malaria. Even malaria we don't get, which is very good. No? We should find out how. If we find out how chimpanzees don't get malaria, maybe we can also prevent malaria, right? So, although there are similarities, there are a lot of differences, which is why we are human. They also don't develop dementia. You all heard of Alzheimer's disease, right? Which is memory loss. Chimpanzees never develop dementia. So don't you think that it's better to be a chimpanzee? You won't get dementia, do you? Yes? Yeah, even I think so. Can I have the next slide? Now, if you look at animals as models for Alzheimer's disease, if you look at aging in animals which live long, like polar bears, they developed something called pointer maybe, which happened, which shouldn't have happened. So what they did was mercy killing, euthanasia of a lot of mentally ill and physically challenged people, and they took those brains because they wanted to study and see what would happen in these patients. Okay? So that was the second largest collection. Now, although it wasn't correct, it gave us a lot of insights into what happens in mentally retarded children. Okay? Now, you have all heard of Parkinson's disease, right? How many of you have heard of Parkinson's? Very good. Okay. So, this is a type of movement disorder. Now, we know that a lot of things has been discovered about Parkinson's. We all know that it is due to a chemical which is deficient in the brain called dopamine, and therefore, doctors can now treat Parkinson's disease by replacing dopamine. Now, how did all this come about? So, it started in 1811 by James Parkinson. He wrote a 51-page booklet on how patients with Parkinson's disease present to the doctor. What are the problems in patients with Parkinson's disease? He wrote it way back in 1800s. Okay? That even till date remains the best description of a patient with Parkinson's. He called it the shaking palsy. We all know that Parkinson's patients have a lot of tremors. It's actually called pill rolling tremors. Pharmacy people may like that. You roll pills like that. No? So the patients with Parkinson's have this kind of pill rolling tremors. They also have drum beating tremors. So basically they have what is called as shaking or tremor. And that was first described by James Parkinson. Then came a pathologist, an anthropologist, that was Rousseau, who first looked at the brain of patients who died of Parkinson's. Okay? So after the patient died, he took out the brain, 1.5 kg brain. He looked at every inch of it and found that a very small part of the brain, which is called the midbrain, this little kind of pointer, right? So this whole thing is called the midbrain. Called mid because it's in the middle of the brain. You can see two black lines here, no? Can you all see it? So that black line is called substantia nigra. Substance nigra is black. So the reason that black line is there is because that area of the brain has cells which is making dopamine. Now if you look at Parkinson's disease, you can see this black is not there anymore. So that is what he described. He said normal people, age matched, have a black line here. Patients who died of Parkinson's did not have this black line here. That's all he said. When was that? 1895. Very good. Then came Louis in 1912. What did he do? By the time microscope was invented, he looked under a microscope and said these are nerve cells in the substantia nigra. This is the brown pigment in it. And within that you can see this pink rounded structure, which he called it as an inclusion. Because Louis described it, we call it as a Louis body. And if we find that, we know that the patient has Parkinson's disease. But it took about 50 years later, 1957, we had to wait for two biochemists to come into the picture. This one here is Carlson, who's from Sweden, and this is Hornikewitz. What did they do? They actually took out that portion, that black pigmented area, tried to find out what the chemical substance is there, which is giving that black pigment, and that led to the discovery of dopamine. Carlson also went on to say that because of dopamine, that is what is going to control fine movement, and if that is disturbed, you have excessive movements. Okay? Now, what did Hornikewitz do? Hornikewitz was the first to try and treat patients with Parkinson's. What did he do? He replaced dopamine. And his first clinical trial, he writes it so beautifully, and I want to read this out. He said the short-term response to the drug dopamine given to a patient was dramatic. What is dramatic? Patients who were bedridden, who could not even get up or stand up when sitting down, who could not move, who could not walk, could do all of this very easily when they were given dopamine. So that tells you that yes, it is dopamine which is reduced in patients with Parkinson's. So if you replace dopamine, you can actually treat. Now you tell me, how did we get to all of these discoveries? What helped? What helped? What's the most important thing that you think led to these discoveries? Come on. First, you all had breakfast now? Speak up. You don't think I would tell? I think it's because of, having the brain available for you to examine. So you have to thank every patient with Parkinson's disease who donated the brain and said, the family said, examine it, look at it, find out what is wrong, let somebody else live. Right? So brain donation is probably the most important. It is the same story with, you all heard of Alzheimer's disease. Now Alzheimer was actually a psychiatrist, he's a German psychiatrist and a neurologist, and this patient, August Dettel, is the first patient whom he encountered with symptoms of Alzheimer's disease. Now he actually followed her up for almost 20 years, and then she died. He had gone to another institute by that time, but he came back because he was burning to know, curiosity. What was it in the brain that made her lose everything? She couldn't even recognize her own husband, she couldn't recognize her children, she had total loss of memory, she couldn't even remember where she was, she couldn't remember where the bedroom was, where her house was, all memory loss. So he was curious, what is this disease? What's happening in the brain? So he came back, he did a postmortem on her and looked at the brain, and this was what you see. So this is a normal brain fluid. Okay? This is grey matter where your nerve cells are, this is white matter. The center holes are called ventricles, that is where a fluid is produced called cerebrospinal fluid. Now compare this with this. Can you see those ventricles are huge? Reason? The rest of the brain is completely shrunken, shriveled up, like a walnut. And why did that happen? Because all the nerve cells in the grey matter had died down. It had died out and got replaced with these tangles, which is a type of protein, and these plaques, which are like coppers. So we know now that those plaques are made up of amyloid, and this led to development of a PET scan, where you can actually detect amyloid in the brain, so that you can make a diagnosis of Alzheimer's disease. Right? So it helps you to know a patient has Alzheimer's disease. So the discovery of that protein is what helped. But you've got to remember that protein alone wasn't enough. We needed to know what is the pathway that leads to this protein to accumulate. And again, it was biochemists who came into the picture, looked at brains of Alzheimer's disease, and discovered that an enzyme called choline acetyltransferase is reduced because neurons producing, like in Parkinson's we have dopamine, here we have acetylcholine. So all those nerve cells died, they didn't make acetylcholine, and therefore, if you look at this area, it's called the hippocampus, that's the memory center. All the brown is where acetylcholine is still there. You can see the top portion there, acetylcholine is gone. It's no longer brown. Acetylcholine gone. In the hippocampus, which is memory center, is what probably leads to Alzheimer's disease. This helped because now doctors are trying to replace with acetylcholine. Problem is setting it into a brain, making it remain there without being broken down. All of that is a big challenge, and that is where pharmacy comes in. Pharmaceutical research comes in. Okay, so that again, all of this discoveries was due to brain donation. Very good. So there are several such discoveries which have been possible due to brain donation. Particularly neurodegenerative, now we know it's all because of an abnormal protein. All the proteins that have been discovered so far are normally present in the brain, but they undergo a change in structure, and we know all that because of biochemical studies on it. So it's actually called a proteinopathy, something going wrong. Okay? So amyloid in Alzheimer's is an abnormal protein. If you want to heard of prion diseases, yes. So there also it is an abnormal protein called prion, which changes structure, and all of these proteins, I've said it propagates. What do I mean by that? Now this dementia is not contagious. I can't get it by touching a patient with dementia. How do I do it? So if one cell, one nerve cell in the brain undergoes a change of the protein to abnormal, it will induce all its neighbors to do that. It's like one of you start talking, all of you will start talking, right? Similarly, brains talk to each other, brain cells. And one goes wrong, everything goes wrong. Okay? So that is how abnormal protein spreads from cell to cell. All discovered thanks to brain donation and biochemists who are working on it. Now, despite all of that, a lot of questions are not answered. There is still no effective treatment for diseases like Alzheimer's. Everybody asks how do we prevent it? We still don't know. So a lot of things are questions remaining unanswered. I put this up because it should inspire you that so many questions are there for all of you to work on. Now this is not just about neurodegenerative diseases. There is also cancer, brain cancer, one of the worst diseases that a person can have. So there are a lot of biomarkers which are coming up thanks to molecular studies, which tells you precision diagnosis, what form of cancer is this. That is something that Dr. Chinmay will want us to tell. What form of cancer? The patient will ask, okay, how long am I going to live? Is chemotherapy that he's going to give, is it going to be effective? All of these questions that you need to answer a patient can only be done thanks to biomarker discovery. Okay, so the first atlas that came out was called the Cancer Genome Atlas, which looked at 20,000 brain tissues. You remember Cushing who had collected? Like that, a lot of people are collecting brain tumors so that these kind of biochemical studies are possible to give you answers. Okay? And this is helping because all of us wear clothes, but I can't wear your clothes, not if you wear mine. You want it tailored to your size. Similarly, every drug that we have to give, we need to know whether that patient is going to benefit from it or not. And that is what is personalized care. This is only possible with all biochemical studies. So back to banking. There are a lot of brain banks across the world. You can see America has 82. You can see Europe has 38. What about Asia? Asia has 60% of the world's population. Do you know how many brain banks we have? How many? I put it up there. There are 4. Very good. And if you look at India, we have only one brain bank. Okay? So you may ask, just blink it now. Because we doesn't be. You ask US to send tissue, you ask Europe to send tissue, let them do all the hard work of collecting, freezing, and keeping it. We just take tissue. Do we need one in India at all? Answer is a yes. Yes, because we are all naked in India, right? So we have certain diseases which are unique to our country. Unique because we still have a lot of consanguineous marriages. And that makes all of these diseases like Lafora body, hot water epilepsy, all of these are inherited diseases and they are common in India. Okay? Now there are also different types of, you would have heard of motor neuron disease. We have Madras pattern of motor neurons. You would have heard of multiple sclerosis. We have an Asian variant of multiple sclerosis. So we can't really use tissue from the West because they don't even have these diseases which are relevant to our country. So there was a brainstorming session in neurobiology in 1985, and that was when it was realized that what's most critical to this country 2-3 decades from 1985 was starting a brain bank, and that is how it began in India. This is Professor Shankar, my mentor. I think a lot of people here do know him. Dr. Jagannath Rao also knows him very, very closely. He was the first to start a brain bank in 1985 to think that this is going to be most important. He fought for 10 years with funding agencies because they didn't understand why should anybody want to back the brain. He fought, he got the funding. Started it in 1995 and now we are 30 years down the line. His objective was very simple. He wanted to provide characterized tissues to any neuroscientist in India who wants to do research. So they don't have to look anywhere, they can just ask and we will provide it free of cost. But what do the families need? Families get to know what exactly they have, and if it is inherited, you would look into genetic counseling. If it is not, good. Right? If it is, let's say, rabies, then we would know that vaccination has to be given to the family, etc. So they also do benefit. So where do we get these samples from? Most of it we take post-mortem at autopsy, particularly in incurable diseases like neurodegeneration. We also take it whenever the surgery is done. So if Dr. Chinmay puts a knife on a patient, we will keep that tissue frozen for research. So what do we do if we take out the whole brain? We have two hemispheres. So one of it we are going to slice like a brain loaf, and each slice will get anatomically dissected out, and we will keep it at minus 86 degrees. Refrigerators at home is how much? We make ice. No? This is minus 20. So this one we will keep it at minus 80. The other half we put it into formalin, which is a fixative to give you a diagnosis. Okay? We also have to clinically annotate the radiology, because without that it has absolutely no meaning. So when we started in 1995, NIMHANS took over it in 2000, and now we also introduced digitization. So we've got 3 decades. You may ask, what have we done? We have moved from 1 freezer to more than 20 freezers, and we've collected so many different brains. From which diseases? We have head injuries, we have infections, we have stroke, we have tumors, we have neurodegeneration, we have psychiatric diseases, and all of these we keep the cerebrospinal fluid as well as serum. We also have biopsy from epilepsy, etc. We keep muscle biopsies, and this is possible because we have round-the-clock autopsy service. So we can't do it at home. It's not like eye donation after that. We have to do it only in a hospital where there is a facility to do a post-mortem. Okay? So how many have we helped? We have people taking tissues from us from across the country. More than 100 projects, and scientists have utilized this, and lots of publications in national and international journals. So I'm just standing here and taking the credit. All the work is done by scientists who have taken the tissues. Just to give you a flavor of the type of studies, you would have all heard of cytochrome P450, I think. We all knew it was in the liver, right? But way back in 1990s, Dr. Vijayalakshmi Ravindra, she asked the question. She was biochemist at that time. She was working at NIMHANS. She said, if you look at patients with psychiatric diseases, some respond, some don't respond. So is there cytochrome P450 in the brain which breaks down these drugs? So Dr. Shankar said, I have the brain, take it and work. And that's what he did. And maybe she was more shocked, but she actually demonstrated that there is cytochrome P450 in the brain. First time in the world. Okay? A lot of other biochemical studies have been done. This is by Dr. Bharat, who's a biochemist in NIMHANS. He looked at protein stability. He looked at oxidative stress. He looked at all of these in head injuries. He looked at it in neurodegenerative diseases. Now we say that mitochondrial dysfunction is there in almost every disease. So he went on to show that it is true. In tissues, there is mitochondrial dysfunction. Okay? He also looked at whether different areas of the brain have differences in mitochondrial function. And if you look at the green, that is the medulla or the cerebellum. And this, across age, there is no mitochondrial dysfunction. Important because medulla is the most important. It has a respiratory center, it controls the heart, and with aging, nothing happens to it. And that's technology, that's God's engineering. He made sure that that is absolutely well preserved. But if you look at frontal cortex, evolved with humans, with aging, you can see that there is a drop normally in mitochondrial function, which makes it vulnerable to neurodegenerative diseases. So in Parkinson's, he had found that Dr. Shankar actually sat and counted the number of neurons. Because at that point of time, it was said that in India, Parkinson's disease was not common compared to the West. So he asked the question, if I count the number of neurons, maybe we have more number of neurons. So he actually did that. He looked at UP, he looked at US migrants, and he compared with Indian. What did he find? He said that there is almost the same numbers. Right? So which means that we should also be vulnerable. But we are not. Why? One of the stories is that we use a lot of spices, we use curcumin, which probably protects us, we don't know. So Sandy was telling me that Guntur is the place for healthy food. So maybe all of you are protected because you use so much spices, right? So that is a story, but it needs to be proven, and you can prove that. We also had Dr. Jagannath Rao. He came to Dr. Shankar way back in the 1990s and said, give me tissue. And he actually looked at Alzheimer's and aging and did this beautiful study where he said there's a transition from beta to zeta helix and there is a defect in DNA repair. That's one of the epoch-making studies way back in the 1990s. We've also looked at several types of infections. India's home to infections, right? And we found that you look at HIV patients, they have a different virus compared to the West. Why is that important? One, vaccine that their West mates should be protecting our strains too. Two, interestingly, our strain of HIV virus doesn't cause dementia. Good, no? We have a very good strain of HIV. Doesn't mean you should get it. We also looked at several other types of viruses. We have helped to people who want to develop kits for diagnosis. Okay? So there is Menhans which developed an HIV testing kit. It's now used by NACO. How do you validate it? You need samples which are HIV tested positive, tested negative, and all of this is in our panel. So if you come up with a kit, come to us, we'll help you validate. We have another Exciton multiplex PCR kit, okay, where we look at 21 different types of infection, one drop of CSF. How do you validate? Come to the brain lab. So this is a very, very precious kit because any child, any adult with a brain infection, you need one drop of that cerebrospinal fluid and you know which infection it is and you can treat the patients. We've also done omics. So when omics hit India and it hit the West, every tissue in our freezers, we gave it to Dr. Keshav and Dr. Akhilesh Pandey, who started the Institute of Bioinformatics. You name the disease and we gave it for proteomics. Did it help? Certainly. It did help us learn a lot. We looked at TB meningitis and found that a lot of proteins which are important for cognitive function are downregulated. So what? If patients survive TB meningitis, they are more likely to develop dementia earlier. Right? So, we also look at rabies. It's one of the most intelligent viruses. Believe me, all bugs are cleverer than humans. Okay? It kills without, and we don't even know how it kills, but we know it's 100% fatal. Okay? So we are trying to look at that. We look at lots of infections in HIV, and for the first time, the human proteome map was put in nature thanks to tissues from the brain bank. Okay? We also looked at epilepsy, a lot of types of epilepsy, particularly hot water epilepsy. What is that? You find it only in South India. We all have this oily bath. We like every Sunday put oil and put hot water. Some patients develop epilepsy with that. So what is this about? So that is something that we looked at, and Dr. Anuranjan Anand, he helped find out the gene for it. We also did a lot of work with IIT Kanpur, and putting all this to tell you that nothing we can do in silos. We have to collaborate with other disciplines as well. So in summary, I think we could say that brand tissues are very, very crucial. One is to understand the disease and the other is to find a cure. Then comes public education. So we have this brain museum that we have at Menhans, and every one of those are brain samples from patients families who donated, who said yes, take it, educate people. You can see Dr. Shankar there doing what he loves most, teaching little children about the brain. He always said we should make everyone N literate. What is N? Neuroscience literate. Okay? So that's what he did. So you can see what he's doing. This is something that he always loved. We've opened out our museum to school children. They come twice a week, and we opened it out to public too, twice a week. And this is really a great fun talking to all of them. How am I doing on time? I can stop. 10 minutes. Okay. So you can all agree now that need of the hour is banks, because you have much more advanced technology than what we started with. And what you need now is tissues. Right? But you would ask the question, okay, if it is so easy, why don't we have researchers lining up at our bank saying, give me tissue, give me tissue? It's not happening, unfortunately. Now to help the researchers, we need 3 people to come together. we need clinicians, we need pathologists, and we need patients to help the researcher. Okay? Unfortunately, if you look at number of autopsies, in 1995 we had 200 autopsies every year. Now do you know how much we have? Any guesses? Is it one figure or two figures? It's just two figures. We're just crossing 20 in a year, and that's not unique to India. That is a global phenomenon. Okay? So autopsies are declining, but demand is increasing because we have so much of omics, etc., etc. And why is it coming down? So we did a little survey. We asked clinicians, we asked administrators, we asked pathologists. The reason is many of the clinicians feel that consent taking is extremely complex. We have a litigious society, everybody going to the court. So they are very, very afraid to ask consent. There is also an assumption that a family has just lost their loved one. How to go and ask the family, give me the baby? But I must tell you that every family that we have approached have never refused. They've only asked one question or two questions. One question is, how come we never knew that this was possible? Second question is, how soon will you give us back the body? No other question. Okay? So, I think if we make sure that we hand over the body soon, they'll never refuse. In fact, today morning 6 o'clock I got a call, there is a family that wants to donate. Okay? And family came forward. Why? Because they heard of it. There is also, traditional tell you the time that I spend convincing a family to go for an autopsy, I can use to save somebody. Fair, but still, he or she is not going to help in science in the future. There is also now virtual autopsies. The clinicians will say, I know the diagnosis, why do I need it? We have advanced technology in imaging. I don't need anything for a diagnosis. We are not talking only diagnosis. We are talking about all those questions we asked in the beginning. Can be prevented, can be treated, can be predicted. How are we going to know that unless you have to show your hand? Now, what about administrators? For them, hello, this is extra expenditure. They are not going to get any revenue from it. An autopsy, you cannot commercialize. You don't pay the patients, you don't take money from researchers. Okay? And there is a fear of litigation. If we find something the clinicians hadn't thought of and the patient could have been saved, I would rather not know. What about pathologists? If all this happens, are pathologists interested in knowledge? Unfortunately, no. Nobody wants to do that extra 3 to 4 hours in a smelly, stinking mortuary, taking out the brain or taking out organs just for somebody to do research. So mindset is something that needs to change. So even if you have clinicians and the pathologists convinced that this is important, what about the patients? What do they want? What do the families want? Now there is a lot of misconception. One of them is that religions forbid autopsy. Answer is a huge no. There is no religion that forbids autopsy. In fact, every religion says don't need. It is the greatest gift that you can give anybody. So the only thing family is worried about is logistics. We want to cremate or bury the body, give it back in time. So this is a group of sannyasis who came from an ashram to the brain bank. I really thought they wanted a golden brain in their hand. But believe me, they spent 3 hours with me, held the brain, and I was trying to teach them anatomy. Instead, they taught me. And everything of what I said was written in the scriptures according to them. So what we are learning is just relearning. There is so much already there. We also went out after signing a brain donation form, saying, take it after I die. Okay? So there are also a lot of social factors and a lot of reluctance amongst clinicians, which I just told you about. Thanks to this, we have missed a lot of opportunities. This is a slide that I take from Dr. Shanta's presentation. He would always say, we missed so many opportunities to learn. So when the COVID-19 pandemic came, we didn't hand back. We did autopsies and tried to help researchers to do research. So let's say the family also agrees. Isn't that the best formula? Unfortunately, what about the researcher? The researcher honestly doesn't want issues. Why? There are a lot of issues. Because you must remember that whatever donations we are having of neurodegenerative disease or anything is end stage. After that, they want to know what happened to the beginning. Right? So, to answer that, there are cohort studies where people sign up for donation during life. And the best example is the nun study of aging. And these are 670 this Catholic sisters from the Sisters of Notre Dame in France who signed up and said, examine me, follow me, do imaging, do cognitive studies, when I die, take my brain. Right? So this was a very, very precious cohort where we actually understood what happens in aging and how it is different in dementia. Now, of course, you have lots of other religious sort of studies, and I must say that that was a gift of love. We have our own celebrities. We have all of these people. I put up pictures of families who said, publicize it. Let everybody think about it and think of donating to a cause. All of them are teachers. All of them continue to teach long after death because they have donated their brains to the brain bank. This is Dr. Kanchan Mukherjee. He's a neurosurgeon. He was working at PGI Chandigarh. Right from day one, he would say, take my brain for research, take my brain. And Dr. Shankar would always joke that, Kanchan, you're going to live longer than me. Unfortunately, he had come to Bangalore for a conference. He died choking on a chicken bone. His wife came down from Chandigarh and said, take his brain. He had wanted that. So his brain has been taken for research because he wanted that. This is Professor Shankar I told you about. He donated his own brain to the brain. Okay? So his legacy lives on. But you may ask, where do we go from here? Autopsies are on the decline. And world over, they've created networks. So shouldn't we also do that? So Sri Lanka, this is Dr. Anil De Silva. He came to Dr. Shankar and said, I want to start a bank. He helped him start a bank. I went to Asia saying, hey Asia, we all pool resources because China, Japan have supracentenarians. How could they live beyond 100? Can we learn from them? They were all very happy to say, yes, let's create a network. What prevented it? Yes? The government. So until government say yes, we have to wait, but everybody is doing it. So I came back to India and said, okay, at least places which are doing postmortems, can we ask them to preserve samples so that all of you can do research? And ICMR helped me start 2 other brain banks, one at PGI Chandigarh, one at AIMS Bhubaneswar, and now we have our own Brain Bank Network India initiative. Okay? So this is the roadmap for the future, improve autopsy rates so that all of you can do research. More importantly, remove waste baskets from hospitals. Whatever is taken out of a tissue, be it blood, be it tissue, be it CSF, bank it. Bank it for who? Bank it for all of you so that it's available for future. Because you are coming up with so many omics. Be it genomics, metabolomics, proteomics, epigenomics, transcriptomics, it's got to pronounce, but you have huge amount of ammunition. So, what or who is going to be the hero of research in the future? Do you think it's clinicians? Do you think it's researchers?
It's the entire ecosystem.
Huh?
It's the entire ecosystem.
More important is, I think it's the biospecimen. That is going to be, and biospecimens can be all or any of these, and you should of course make sure that it is fit for purpose, which means it should be the right specimen collected at the right time, in the right way, the right test chosen for the right result. Okay? So tissue banking is going to be building blocks. It does not have to be post-mortem. It can be any tissue that is taken out at surgery, because precision medicine is going to be the priority is. Okay? And you have a lot of advantages, you have a lot of advances with omics, machine learning, blah blah blah, which can answer all of the questions we started. Who's going to answer the questions? All of you. All we have to do is an easy job of collecting samples and giving it to you. But the banks are going to be useful only and only if all of you use those samples for research. So, the unsung heroes in any kind of research is going to be the biospecimens, and I must tell you to stop working on those poor mice and start using human tissues. And leave with this very small poem. This is written by the father of a young boy who died in an accident, and then his body was lying on a stretcher covered in white cloth. This is what he wrote. He said, explore every corner of my brain, take every cell so that someday a speechless boy will shout at the crack of a bat and a deaf girl will hear the sound of rain against her window. Give my sight to a man who has never seen a sunrise or a baby's face. Take my bones, every nerve and muscle, and find a way to make a crippled child walk. If you must bury something, let it be my faults, my weaknesses, and prejudice against my fellow men. So brain donation is probably the greatest legacy that we can leave as a gift of hope for generations to come. I want to end by thanking this entire group without whom none of this would have been possible. Clinicians who took that extra time to convince the family, the families and donors, Nimran's administration for saying go ahead and do it, although Nimran's is getting nothing out of it. Our mortuary and brain bank staff who are with me 24/7, anytime, day, night, they will say, come, let's finish the house. I'll say, no, let us do it in the morning. No, we have to do it now. That's commitment. During that COVID-19 pandemic, I was hesitating because vaccine had not yet come at that time. But my mortuary staff said, madam, you have done prion-disease, you have done anthrax, you have done rabies, you have done all that, you're still alive. Why don't we do COVID-19? And we did it. Okay? So that's thanks to people like that. And the master of all, of course, Professor Shankar. And this entire talk is dedicated to this one man who thought that it's most important for this country to have a brain bank. And what we are trying to do is just continue that legacy of his generosity of giving. Because that's the mental mindset we need to have. Many of us have projects, we will collect samples, we think to ourselves, but if you ever think of giving to others, that is a mindset that Dr. Shankar has inculcated in all of us, and we are trying to continue this legacy. Thank you very much for your attention.
About brain banks and several neurological diseases like Parkinson's and Alzheimer's. Not only about neurological but also about brain cancer and all. Thank you, ma'am. It is very much important for many students like us who are more interested in neurological studies. Thank you, ma'am. Thank you so much.
And we are so privileged to have you here as a part of our international symposium. Thank you, ma'am. And now, it is a golden opportunity to the students present over here to grab this opportunity and clarify your doubts or queries regarding to the session given by our Dr. Anita Mahadevan, ma'am. So please kindly raise your hands and clarify your doubts.
Different types of biomarkers. In fact, I think maximum data comes out of these kind of studies. Validation of which biomarker is best is what is most critical. So it isn't one biomarker that you can tell. You usually get about 10,000 plus biomarkers. What is important is finding out which is most useful to answer your question for that particular disease. Okay? So you have a lot of work to do. Yes? You can all come up to a picture in my cup.
Actually, the current trending topic is artificial intelligence. How artificial intelligence changed the way of neuropathologists, change the analysis, analysis the brain tissue.
A very good question. In fact, I think they're all the time fighting which is better, human intelligence or artificial. But what AI helps you to do is, like she was asking about biomarkers. You have so much of information. To make use of that to answer a question, to be able to use big data. We have a lot of data which needs to be analyzed. I think AI can help you do that. That is beyond human intellect. We have a capacity only. But if you want to put multiple data together, like if you look at Alzheimer's, I want biochemical data, I want radiology data, I want clinical data, I want the cohort data. If you want to put all of that together and find a meaning, that is where I think AI is most useful. But getting the data is not from AI. That is HR. What is HR? Human intellect. Does that answer?
But actually, you mentioned about brain banking. How brain banking helps in the finding of biomarkers in Parkinson's?
Yeah, good question. If you saw my slides, initially what did we have? We had clinical, means we knew how a patient with Parkinson's presents. Then we had the brain tissue to be able to look under a microscope to say, if you find Lewy bodies, this is Parkinson's. Then came, what does that material contain? We knew it was synuclein. Now we know synuclein is there normally as well as in Parkinson's. So what is the change that happens? Like Dr. Rao showed beta to zeta helix. Like that, protein changes configuration. We knew that thanks to availability of tissue. But what is it that converts it? We need to do more studies. That is where biochemistry comes in. So at every step, one, we need tissue. But remember, any tissue that we put into a bag, it's called G goes, isn't it? Garbage in, garbage out. So if I wrongly diagnose Parkinson's and give you tissue to study, you get wrong results. So the most important is to first characterize the tissue. Make sure that the diagnosis is correct. Right? And only then give it for research. So this tissue is available. You can do all kinds of omics. You can tell what happens at protein level. You can go up there and look at what mRNA level happens, what happens at DNA level, what happens at epigenome level. So all of that can be done, provided one thing is there, well characterized tissues which are frozen. Okay? So that's how banking happens. Somebody has to bank so that all of you can do. And you may also ask me why only Parkinson's patients should donate. No. We need age-matched controls also. Okay? You have to compare. Everything is comparison. So if you have controls only, you can say that compared to this is the abnormal. So you need both patients. One more question. She allowed three?
No.
Let the others ask, then you come back. Yes?
One ma'am.
Yeah.
What are the other innovations could be improve the brain banking and accelerate the neurological research?
I need another hour to answer that question. It's a very good question. But that's what I was trying to summarize there. One, we need people committed to this banking process. For this, we need clinicians, we need pathologists, we need researchers wanting to use that machine. So most important, I would think, is all of us should consider banking as the most important for future studies. And banking is only not after death. You get bank blood, you get bank CSF, you get bank. Now people are even looking at tears and saliva to diagnose Parkinson's disease. Right? So how can we validate all that? We need samples from patients who are frozen. We need control. So any sample from a human being is worth preserving. People are looking at students. They want to look at microbiome and saying that that is what is going to dictate whether a person develops Parkinson's or not. So you need to bank students.
Good morning, ma'am. During child age, we observe childish behavior in children, and at that child becomes old age, we observe childish behavior in also them. What type of changes did we observe? Brain changes during child age to old age in them.
Questions are getting more difficult, sir. Good question. So, okay, I'll hide behind the statement that what we know about neuroscience is only tip of the iceberg. There's a lot that we don't know. We can understand structure with all of these studies, including what we are finding difficult is function. So all of us come with the same kind of nerve cells, networks, etc. How does that same network you have, same network I have? You think of wonderful questions like this, I cannot. So how do we all differ is a question. So now there is a lot of studies which we are trying to use to understand mind and behavior. We are moving from brain structure to function. Now, what changes after? There are a lot of imaging studies now which are trying to look at, we call it connectomics. This is a study which was done by Dr. Vivek Benegal, who is a psychiatrist from our institute. His study is titled From Cradle to Grave. Okay? He has this long cohort of patients whom he follows up, right from birth to death, trying to understand behavior. So we all say that it's the second childhood with old age, right? We don't know what changes happen. We can't explain it by saying protein change is similar from child to it is not. So there is something more, and that is where connectivity comes in. Networks connect. So he's trying to understand that. I think if we give him another 2 years, he's collected enormous amount of imaging data. Enormous amount. And this is not structural imaging. He's trying to look at functional imaging, which is you would have heard of functional MRI, you would have heard of PET scans. So this he has done for a person from birth. So lot of psychiatric diseases we are not able to understand because there is no structural change. So that is why these kind of studies are important. But like she asked me, big data, we need AI to analyze that kind of data. So short answer to your question, we don't know. But a lot of studies are going on to understand. Okay.
Thank you.
All of these neurotransmitters, there is a clusters. Increases and decreases, even normally. For instance, it controls our mood. You like chocolates? If you have a chocolate, you'll feel happy. So what is happening there? Dopamine is increasing. There's a surge. So each nerve cell which makes dopamine has cues. So if you eat a chocolate, you feel happy. If you speak to your friend, you feel happy. If you see your biggest enemy, then what happens? You feel sad, you feel anger, dopamine is coming down. So every nerve cell has an on and off switch, which has cues. So we can't tell that there is a cutoff, like blood sugar, more than 200 is bad. We can't say that. So it's going up and down. But beyond a particular threshold, it causes disease. So excess dopamine will cause schizophrenia, will cause mania, psychiatric disorder. Very low, beyond the threshold, causes Parkinson's. So normal also there is a flux.
Good morning, ma'am. I want to know the answer from our students as a question of you. How many young brains are ready to donate the brain for your brain bank?
Is a question you are asking?
Yes, ma'am. I
Raise
want
your
to
hand,
know.
how many of you are inspired? Oh my.
How many of you are ready to donate your brain towards the brain bank? How many brains are ready?
I rephrase that question. Right? So I want to know how many of you are interested in research on all. Right? There is your answer. Thank you for thinking about the nation. Think about it. That's enough. No more questions?
Any more questions?
Thank you.
Thank you, ma'am. Thank you for patiently answering and clarifying the doubts from our students. Give a big round of applause for our Anita ma'am. Now I request Chairperson Mr. Saiba Hussain to kindly come onto the dais for addressing the closing of this session. Give a round of applause for the
Good morning, Manila. Another nice session, madam. It's my privilege to briefly highlight the implementation, concept, and discussion lesson of brain banking in India. Its legacy for discovery, innovation, and hopes. Brain banking is a valuable scientific resource that provides well-preserved human brain tissue for advanced research. It plays a crucial role in understanding complex neurological disorders such as Alzheimer's disease, Parkinson's disease, and other neurological conditions. The presentation emphasized the study human brain tissue, helps researcher understanding the diseases, mechanism at the molecule level. Many discoveries related to the protein pathosis have emerged through the brain banking. For example, Alzheimer's disease. The speaker also highlighted the limitation of traditional animal models. Although animal provide useful information, they do not completely reproduce human brain disease. The new approaches such as human brain tissue studies, molecular biological technology, homeostasis technologies, and stem cell based models. Thank you, Ananda.
Thank you, sir. Thank you for closing the session. It is my privilege to express our heartfelt gratitude to the esteemed speakers and chairpersons here. And now I invite all our dignitaries, Secretary Ma'am, Advisor Sir, Global Secretary, Dean of Academics, Principal Sir, and all the faculty members to kindly come on to the dais for felicitating our chairperson, Sri Sai Babu Sir. I request everyone to join your hands and give a warm round of applause.
ఎందుకని ఇలా వెనక లైటింగ్ ఎక్కువ తగ్గిమని చెప్పండి కొంచెం ये उठाता तो इसका ये दिसना ही अरे गोरी होती है दो राशि दोनों बात कर
give him a round of applause.
సార్ కొంచెం బ్యాక్ ఓకే సార్ మేడం
పిచ్చే బ్యాక్గ్రౌండ్ ఫుల్ వైట్
Thank you, madam, and thank you, sir.
ఫోకస్
It gives me immense pleasure to welcome Dr. Kavita Babu, Professor at IISc Bangalore. I request the Chairperson Dr. J.N. Suresh Babu, Principal of Naturopathy Institute of Pharmaceutical Sciences, and Co-Chairperson Dr. Raghuveer, Indo-American College of Pharmacy, Journal Letter Naturopath, to graciously introduce our distinguished speaker and share highlights for her inspiring career and accomplishments before moderating the session.
I request everyone to give a warm round of applause on welcoming our guests onto the dais. Your energy is what matters in our international symposium.
Good morning, everyone. And it's my privilege to read out the biodata of the chairperson for today's session. And it's my pleasure to read out the biodata of Dr. J.N. Suresh Garu. Dr. J.N. Suresh sir is an eminent academic administrator, physical educator, pharmaceutical educator, and researcher with over 3 decades of distinguished experience in pharmaceutical education, academic administration, research, and institutional development. He is currently serving as a principal and professor at Naturopathy Institute of Pharmaceutical Sciences, Naturopathy, where he has been instrumental in strengthening academic quality, research culture, and professional excellence. He completed his B.Pharm from Gulbarga University, M.Pharm from Mangalore University, and obtained his PhD in pharmaceutics from Jawaharlal Nehru Technological University, Hyderabad. Dr. Suresh Kumar possessed extensive teaching and administrative experience, having served in several reputed pharmacy institutions across Andhra Pradesh and Karnataka. During his illustrious career, he has held key academic leadership positions, including assistant professor, professor, principal, and professor at institutions such as NGSM Institute of Pharmaceutical Sciences, Mangalore, Bharati College of Pharmacy, Mandya, ML College of Pharmacy, SGIPS, Markapur, Deccan College of Pharmacy, Hyderabad, and presently at Naturopathy Institute of Pharmaceutical Sciences. His research interests include pharmaceutics, drug delivery systems, pharmaceutical education, pharmaceutical jurisprudence, and forensic pharmacy. He has published several research papers in national and international peer-reviewed journals, presented scientific posters at numerous conferences, and authored the textbook Forensic Pharmacy, which serves as a valuable resource for pharmacy students and educators. A committed professional leader, Dr. Suresh Kumar is a life member of IPA, Vice President of APTI, Andhra Pradesh State Branch, Life and Executive Committee member of the Indian Pharmacy Graduates Association, Andhra Pradesh State Branch, and Associate Life member of Indian Hospital Pharmacists Association, and a registered pharmacist under the Pharmacy Act. Known for his visionary leadership, academic integrity, and dedication to quality pharmacy education, Dr. J.N. Suresh Kumar has mentored countless students and faculty members while promoting research, innovation, and ethical professional practices. His unwavering commitment to excellence continues to inspire the next generation of pharmacy professionals and contribute significantly to the advancement of pharmaceutical education in India. With these remarks, I may now invite Dr. J. Suresh Kumar Garu, please. Sir, please.
Thank you, Ravit, for great introduction. Friends, dignitaries, invited guests, and my dear students, I am very happy to be here on this international conference on brain and disorders. And it's my privilege to introduce today's eminent speaker, Dr. Kavita Babu. Dr. Kavita Babu is presently a professor at Center for Neurosciences, Indian Institute of Science, Bangalore. She has completed her PhD in Cell and Molecular Biology, National University of Singapore, King's College London in 2004. She is a professor presently at IISc Bangalore. She is a postdoctoral fellow in Harvard University from 2005 to 2011. Her research is mainly focused on synaptic cell adhesion molecules, neuropeptic signaling, and molecular mechanisms of learning the long-term memory using C. elegans. She has 20 plus publications including PNAS, communication biology, genetics, and developmental stuff. She is a fellow of National Academy of Science, India. She is the Indian representative for PRO. She is the recipient of major competitive grants from Department of Biotechnology, ANRF, SERB, and Welcome Trust India Alliance and Ministry of Education. She is the organizer of major international neuroscience conferences, and it's our privilege and great privilege for me to introduce her at this juncture. Friends, neurosciences is picking up its importance in the field of medicine. In every family, we find at least one person who is suffering from neurological disorders, and it is the alarming situation, I can say. One has to be careful right from their teenage, or by changing their lifestyle modifications. One has to be free basically from stress. As students, you all will be having a lot of stress, majorly for gaining marks, scoring. Beyond this, so many stressful situations every student is facing nowadays. Very simply and very silly things. The student will feel more stressed if he or she does not get more likes in their Instagram post. A student wants to be a point or center of attraction, which is called as spotlight syndrome in every class. If they don't speak, if they don't interact, if he is ignored or she is ignored, they feel a lot of stress. Friends, please concentrate only on studies. Please have in mind what is your motto and work for it, and try to have a little bit of calmness or meditation in your day-to-day life. Then I'm sure you all will be free from these neurological disorders with good health and good meditation. Thank you very much. I hope I'm right, madam. I request the speaker of today, Dr. Kavita Babu, on to the dais. Handing over the dais to madam. Thank you.
Thank you for the very kind introduction. And the previous speaker had talked very nicely about brain banks, and today I'm going to go way down from human brains and talk about model system called Symposium elegans, which is allowed for multiple discoveries in, especially in the realm of molecular biology, and its simple nervous system has allowed us to study a lot of molecules in neurons in this organism. Is there a pointer? What can I do? I can change something. I can change that. Alright. So, all of you know that the human brain is one of the most so the human brain is one of the most important organs in our body. It's required for us to walk, talk, think, eat. Everything requires the brain. And it's made up of multiple cells, which are called neurons. And these neurons talk to each other. One neuron can talk to multiple neurons, and they go on to talk to more neurons. And this is how essentially we function. So why look at Symposium elegans or C. elegans? As we know, the human brain has around 100 billion neurons. C. elegans has exactly 302 neurons. So that makes you actually be able to look at every single neuron very easily. Humans have more than 10 trillion synapses. C. elegans has approximately 7,000 synapses, again making it easier to look at synapses. However, when you look at the molecular level, humans have around 25,000 protein-coding genes. C. elegans has around 20,000 protein-coding genes. And of these genes that are required to act as neurotransmitter receptors, genes that are required to act as transcription factors, genes that are required to allow for making enzymes that make neurotransmitters, most of them are conserved in C. elegans. So around 60 to 70% of the genes that are in humans are also present in C. elegans, and especially all the genes that are required to make dopamine, serotonin, acetylcholine, all of them are conserved as other receptors for these neurotransmitters. So this then allows us to come down arrow. Oh yeah. Yes. Thank you. Yes. So now this allows us to start thinking about how we can look at molecules in this model system, and this is how we look at it. So C. elegans is essentially a transparent organism. So you need a microscope with light at the bottom and light from the top, and that will allow you to look at it. What you're seeing here is essentially different stages of C. elegans. The asterisks are the eggs. So C. elegans is largely hermaphrodites. So they have both the male and the female sex organs in the same animal. And this single animal sends out eggs, and all of those eggs are exactly the same as the mother. Apart from that, you also have male C. elegans. So you have mating between hermaphrodite and males, and that's how you can do a lot of very interesting genetics in C. elegans. So what we have here are the eggs. You see the small guys around, those are the N1 stage of larvae, and I'll tell you a little bit more about these stages. The little bigger ones are the N2 and N3, and the large ones are the adult C. elegans. And what do they feed on? Essentially, I know if you guys have seen bacteria, you've seen a plate where you where you streak bacteria. Yes? Yes? No? Yes, right? So essentially C. elegans eats bacteria. So you just take a plate with agar and you just put some bacteria on it, and the worm can sit on that plate for a month or more. And so it's very straightforward to work with C. elegans. And so this is the life cycle of C. elegans. You have the hermaphrodite adult, which gives rise to the egg. The egg is on the plate. It goes through a life cycle and goes through 3 different larval stages, four different larval stages, N1, N2, N3, and N4. N4s then give rise to adult. And this whole process takes only 3 days. So essentially, you're working with an organism which has neurons at the speed at which you're working with yeast. Yeast takes around 2.5 days to propagate. This takes 3 days to propagate. around 250 works on that. So with this, I'm going to tell you a story that we recently published from the lab, and the story essentially deals with swarming in animals. I don't know if you guys have seen these YouTube videos of these starlings which form these swarms. They all come together and form these beautiful swarms of animals. This is essentially a clip from that, a small bit from that video. You have these starlings all coming together. But apart from starlings, locusts also form swarms. Multiple types of animals form swarms, and as they come together. And we wanted to, actually, we did not want to study this in C. elegans, we wound up studying this in C. elegans. So I'm going to tell you how this process happened. And all of this work was done by a graduate student, Navneet Shahi, who is going to be leaving the lab in a month or so. And what you're seeing over here is a C. elegans that's moving around. So she has worked with undergraduates. She trained 3 undergraduates during the time she was in the lab, and all of them have contributed to this one. But this is her idea, and the thoughts are all hers. Okay. So, just to kind of reorient you again, this is essentially just to show you the size of the animal. Okay? So, a C. elegans is approximately, the adult C. elegans is approximately 1 millimeter in length and 50 micrometer in width. So that's the size, and you essentially look at it under the microscope. So this work started many years ago actually in our lab, when a graduate student, Shruti Thaplia, was interested in looking at this gene called calcitonin. Now calcitonin is a very conserved gene, and it's conserved from C. elegans to humans. And mutations in this gene have been implicated many different neurological diseases like autism, Alzheimer's disease, schizophrenia, bipolar disorders, etc. And people have essentially done studies where they found that there are certain point mutations in this gene that might be involved in these diseases. So, Shruti wanted to study what happens to this gene in C. elegans, and she was my first PhD student. And what she found very nicely was in C. elegans, unlike in humans. So in humans, you have 3 independent genes, through 3 independent calcitonin genes. In C. elegans, you have a single calcitonin gene, which is spliced to give 3 independent isoforms. And what you found very interesting was that, I don't know if you can see this, okay, so in the left side, you're essentially seeing calcitonin is largely expressed in neurons. It's not expressed in many other tissues. And on the right side also, you just see that the 2 other isoforms, B and C, are essentially expressed in the ventral cord neurons. So this calcitonin, Cas1, is largely expressed in neurons in C. elegans. And it's the same with CST1, CST2, and CST3 in humans. They're largely expressed in the brain. CST1 is minimally expressed in other tissues, but CST2 and 3 are just expressed in the brain. So with this information, Shruti went on to study what happens, and she found that mutants in this calcitonin reduce GABA, the neurotransmitter GABA, released from motor neurons. Okay, now, this was published some time back, and Navneet then started working on calcitonin. And we were trying to do many different things, but there was one thing that she kept seeing. She kept seeing in the plate that the worms kind of form these clumps along the bacteria. So the circle is the bacteria. And she kept seeing that they were forming these clumps. And she kept asking me that I want to study this. And I'm like, how are you going to study it? We have no idea what's happening. She said, don't worry about it, I'll figure it out. I was like, okay, fine, let's see what happens. So the first thing she did was she made some videos. Now I don't know how to get these videos to work. So you'll have to click on those things. I don't know how. No, previous. See here. There will be, if you move, you'll be able to video play out the video play. Here. Actually. Yeah. So if you worms, they're all put only in one direction on top of the floor. And you can see that these are moving very differently. They're all coming as a swarm. They're all moving across the bacteria as a swarm. So this was the first indication that maybe this gene is required for some sort of swarming. Now I do want to tell you that this is one of the first videos that she recorded. And she had put in more than 1,500 worms in each case. However, when we went on to do experiments later, we did not want to deal with so many worms, so we reduced the number of worms. And this is what the swarming looks like. Can you go again? Please? So this is what these videos are going to do. You can do both of them simultaneously. So this is pure worms. This is around 700 worms, which is half the worms, because we had to do a lot of rescue experiments. We have to pick these worms. It just becomes harder and harder, but you can see that it nicely swarms. So with this information, I was like, all of this is very nice, dummy, but how are we going to quantify it? Any kind of biological, fault that you see, unless you can quantify it, it becomes very difficult to say that there is a defect, although you can visually see it very nicely. So then, what Navneet did was, she used a quantification scale, which is called relative dispersive, which is just a grayscale analysis. She used this using a plugin in MHA, and she just did a quantification of that. And you can see that wild type animals show higher relative grayscale, while the mutants show a lower relative grayscale. That's because in the mutants, all of these guys are coming together. In wild type, they're all more dispersed. She also did another experiment. She put back the single gene into the mutant, which did not have this gene. She put back the gene, and she found that she could almost completely rescue this phenotype. So putting back this one single gene into the worms could rescue this phenotype. So this allowed her to say that okay, we see this mutation, we see this phenotype where the worms are swarming together when one single gene is lost in these worms. So now I'm going to just tell you a little bit about what are the experiments that she initially did, and then we go on to see what Cassipon is actually doing in this system. So when she started, she looked just like Shruti did. She looked at the entire Cassipon. This is the full A isoform, or the largest isoform of Cassipon. And what Shruti had initially got was this small deletion in Cassipon, this TF71. Now what Navneet did was she took CRISPR-Cas9 and she made a complete deletion. That in 107, deletion is made by her, where she completely deleted the Cas1 proteins. She also found another deletion available from another lab, which had the same kind of complete deletion. She looked at that as well. All of these three mutants look very similar. They all show this swarming phenotype. And putting back Cas1 into any of them completely rescued this phenotype. So all of this gave her more encouragement that she's in the right direction. Cas1 must be causing this swarms. So her next question was, in which neurons is Cas1 required for this swarming phenotype? Now I had shown you previously that the A isoform is expressed in the head of the worm, while the B and C are largely expressed along the body of the worm in what are called motor neurons. So what she did was, she put back Cas1A wherever it was. So she used Cas1A promoter, put back Cas1A, and she could completely rescue the phenotype. She put Cas1B with the Cas1A promoter, again she could rescue the phenotype. However, when she used a Cas1B phenotype, it's not expressed in all these head neurons, but expressed more in the ventral cortex neurons, she could not rescue. So this indicated to her that Cas1 is required in the head of the worm. So you put back Cas1 in the head, the neurons in the head of the worm, you can rescue the phenotype. You put it back anywhere else, you're probably not going to be able to rescue the phenotype. So this allowed her, so she had some location of where Cas1 is required to cause this swarm. So now, what could be causing this swarm? Now, obviously, one of the first things we thought, when you look at these worms that are swarming, one of the first things we thought was they're secreting something that is allowing other worms to stick to them like a glue. Okay? One of them is called pheromones. Now pheromones are sent out by worms. I told you that worms you can have hermaphrodites and males. So mostly males send out pheromones that attract hermaphrodites. You can also have pheromones sent out by hermaphrodites. So we decided to see whether pheromones were involved in this process. The second thing we wanted to look at was extracellular vesicles. Again, a large amount of work in extracellular vesicles is extracellular vesicles are required for mating. Again, both males and hermaphrodites are called these extracellular vesicles, and these are required for mating. So we looked at mutants that prevent pheromones from being formed and other mutants that prevent extracellular vesicles from being formed. And what you found was nothing. Mutants that did not have pheromones, so you have DAC22, which is defective in pheromones, swarm exactly like wild-type animals. KLP6 is defective in extracellular vesicle release. They also swarm exactly like wild-type animals. So it was not because of release of pheromones on extracellular vesicles. Now, we could not think of what else was released from the worm because we don't know. So at this point we said, okay, let's just leave it at this. She also looked at one other thing, and that was odor. So another process of this swarming is very specific. It happens only when there's food. So you need this patch of food in order for the worms to swarm. So she decided to see if they cannot sense the food, will they still be, would they by any chance give a swarm instead of going like wild? And the answer is no. So she had mutants which did not sense food. They do not have odor receptors, and they do not swarm. They look just like food. So all of this basically indicates that it's not a sensation towards food. It's not something being released, or it's not at least pheromone or EVs being released that is giving rise to this food. So what could be the next thing? Could it be intrinsic factors? Could it be neurotransmitters or neuropeptide? Or if you've heard of neurotransmitters, you've heard of dopamine, you've heard of serotonin, you've heard of acetylcholine, GABA, all of these are known neurotransmitters. So what she decided to do was, she decided to look at, she decided to look at mutants of all of these known neurotransmitters. So these are known mutants that affect these neurotransmitters, and by effect, essentially most of the neurotransmitter is not released. There's very little neurotransmitter. She looked at glutamate mutants, GABA mutants, acetylcholine, dopamine, serotonin, octopamine. None of them showed a phenotype. So there was no swarming in any of these mutants, indicating it was probably not the loss of any of these neurotransmitters. So this is what they do. They look just like that. So then, at this point, I was at a meeting, and one of my colleagues, Raghav, showed me this paper, and he was like, maybe it's because what you're saying is seen in locus, and maybe it's because of increased serotonin. So we thought, okay, maybe it's because of increased serotonin. It's not loss of serotonin, it's increased serotonin. And I don't know if you've heard of this neurotransmitter called serotonin. Serotonin has been implicated in many different functions. One of it is for being related to eating, related to food ingestion, and knowing how much food to eat. What she did was she supplemented the plates with serotonin, and she did start seeing a small formation. However, I do want to say one thing over here. The mutants, Cas1 mutants, show a small formation that nothing else that we have seen can replicate. They chose a very, very clear cut and very, very, obvious swarm formations. All the others show a little bit of a swarm formation. It's not as great as a Cas1. So we thought, okay, is this swarm due to excess serotonin that would be in Cas1? So that was a question that we asked. And what we did, what Naveen did was essentially add serotonin to the plate, and you can see that adding serotonin to the plate showed a small-like formation. While if you do the same thing, you take Cas1 mutants and add serotonin, it does not increase the swarm formation. So this indicated that Cas1, at least a part of that swarm formation, is probably due to excess serotonin. So now, if this was And that's exactly what she did. So just to give you a brief idea, you all would have heard of amino acids? Yes? Yes? You heard of the amino acid called tryptophan? Yes? Right. So you have tryptophan through an enzyme called TPH1 gives you 5-HTP, which is the precursor to serotonin. So, the next enzyme BAS1 converts 5-HTP to serotonin. So now you have this serotonin synthesis pathway, and this serotonin is then released, and it does two things. One, it binds to a receptor which is on the next neuron, which is in CLS, it's called the MOD1 receptor. Okay? It also binds to a receptor which is called a reuptake receptor. The same neuron has a receptor which will be serotonin will bind and allow this process to continue. So now, if you want to remove TPH1 or BAS1, you have less serotonin. If you want to remove MOD1, you have less serotonin. But if you want to remove MOD5, which is the receptor which is required to sense serotonin and start this pathway, you will get more serotonin on the cell. Okay? So using this information, now we looked at BAS1 mutants, they look just like wild type, but when you couple Cas1 and BAS1, you go back to wild type. So BAS1 is less serotonin. Let's assume Cas1 is more serotonin. The less serotonin will win, and you go back to this. She also did MOD1. Again, MOD1 mutants will have less serotonin, or they do not sense serotonin. You make Cas1 and MOD1 double mutants, go back to wild type. You no longer see the increased serotonin. Now the third thing she did was just look at MOD5 mutants by themselves. MOD5 mutants have more serotonin, and what she sees is MOD5 mutants look like Cas1, again, not as nice as Cas1, but close enough to Cas1. So all of this allowed her to say that probably what we are seeing is because of increased serotonin that's there in Cas1, but it may not be the whole story, there may be other things that we don't know. So now, if this is going through serotonin, can we figure out who regulates this pathway? Okay? So what are the regulators of this pathway? What is known as that? You have few neurons that express serotonin and C. elegans. All these neurons are present in the head of C. elegans. And all of these neurons also have Cas1. So these neurons express both Cas1 and serotonin, so it's a good place to start. Now, previous work has very nicely shown that you have a pathway that involves a neuropeptide. Have any of you heard of what is a neuropeptide? Yes, no? No, say no. Okay, I'll assume it's no. Essentially, you have neurons which send out these small peptides, and these can be thought of like hormones in some ways. For instance, C. elegans sends out a number of insulin-like peptides, which is similar to insulin hormone in humans. So these peptides then bind to receptors which are on other neurons, and they can activate or inactivate those other neurons. So it's essentially very similar to neurotransmitters. The only difference is they can act on neurons when they are not synapsing on it. They can act on little far away neurons. And the second thing is they act slowly, and the action stays on for an extended period of time. Neurotransmitters are very fast acting molecules, but neuropeptides are slow acting. Okay, so with this, you have a neuron that's what previously people have shown is that serotonin is regulated by this neuropeptide called PDF1 and its receptor which is called PDFR. And what is known is that if you have increased serotonin, you have less PDF1 and less PDFR1. You have decreased serotonin, you have more PDF1 and more PDFR1. So they are inversely related to each other. So this allowed her to now look at this peptide. So she wanted to see, is it the same peptidergic signaling that affects serotonin also works with Cas1? And the answer is yes. You look at PDF1 mutants, they look just like Cas1. You look at double mutants, Cas1, PDF1, look like Cas1. PDFR1 mutants also look like Cas1. So now what she did was she thought, okay, if this is the case, then if I were to just put in PDF1 through these worms, give these worms a lot of PDF1, can I rescue the phenotype? And that is indeed the case. You can see PDF1 put back PDF1, rescues just looks like wild type. Wild type put back PDF1, looks like wild type. Cas1 put back PDF1, looks like wild type. So all of this indicated that if you just put back this one neuropeptide, you can completely rescue this phenotype. So this is just like wild type. You're putting back PDF1 and Cas1, it looks just like that. So now, we were in a position where we had a circuit. Now we could actually come up with a circuit where we could say that these neurons might be giving up serotonin, these neurons could be giving PDF1, these could be giving PDFR1, these could be giving Cas1. Now we wanted to test this circuit, and for this we use a technique which is called optogenetics. We were essentially activating or inactivating neurons to see what happens. So now what we thought was, if we can activate, so I told you that serotonin is what is giving, if you have too much serotonin, you're getting this phenotype. Now, that increased serotonin indicates that there is less PDF1 and PDFR. Now, our idea was, if we can activate PDF1 and PDFR1 neurons, we can activate them by some mechanism, we could in fact rescue the phenotype. So in Cas1 mutants, we just activate these PDFR1 neurons, can we rescue them? So we just activate these PDFR1 neurons using black, which is blue light activated adenylyl cyclase, which will allow you to activate specifically just the PDFR1. Okay, turn on the video. And you can see this, it's going to take a little time. So this is Cas1 mutants with this PDFR1 black. And you're going to see a zzz when the blue light is added. Okay? So this is the blue light has been added. And you can see that with each iteration of blue light, the worms stop squatting. They look just like wild type worms and moving everywhere. Indicating that just by activating a subset of neurons, you would completely rescue this phenotype that was there. And this is just looking at all the different units. So we did this with lots and lots of controls because we had to use just the blue light. We had to use PDFR1 without the black, lots of different controls. So here you can see this is before light, Cas1 black and wild type, and after light it's no difference. This is just Cas1 without the black, no difference. After the blue light, you're still seeing the swarm. This is Cas1 black. In Cas1, you can see after the blue light, the swarm no longer exists. PDFR1 black in Cas1 does not affect PDFR1. So all of this indicated that maybe what's happening is through these PDFR1 neurons, Cas1 is affecting serotonin. So now, we decided to go one step higher. C. elegans has only 302 neurons. Can we pinpoint that one neuron that's involved in this process? So now we went extensively to look at what each neuron expresses. All of this is there. We have databases of a lot of transcriptomics, which happens in every neuron. We know pretty much what every neuron expresses, what every neuron does not express. All of that is known. And what she found was there was this one neuron called the ARY neuron. This expresses Cas1, it expresses PDFR1, it expresses MOD1, which is serotonin receptor, and it also expresses PDF1 and Cas1. So we thought, okay, let us see if this neuron is enough. This AIY, this one neuron, is it enough to cause swarming? And what she did was she inactivated this neuron in wild type animals by turning on another light. So ARCH is antibacterial, and putting this into the neuron and shining light will cause this neuron to be inactivated. Okay? And you can see that when you turn on this video, both simultaneously, please. Okay. So you're turning on, so basically you're inactivating this one single neuron, and what happens during the inactivation process is you're causing a swarm formation. So this is just wild type animals, you're inactivating it. You're getting this little bit of a swarm formation. It's not a very nice thing to go again. So it's not like the control, the control are not seeing a swarm. In the mutant, you're seeing a swarm formation to some extent. Now we don't think this is the entire story, but we think this might be part of the story. That this one single neuron might in itself be enough when inactivated to give rise to this swarm. Alright. We also did another experiment where we activated this single neuron and we found that the Cas1 swarm is lost. So you activate it using another of the genetic technique and you lose the swarm formation. So all of this indicated that maybe this AI neuron is enough, but it may not be the entire story. We don't think it's the entire story. We think there are many parts to it, but what we found is a very, very small one. So, can you play the video? Right, yeah. So this is just, again, we are just activating that one neuron in the Cas1 matrix. And again, you're going to see the blue light, yeah. And this stops the swarm formation. The swarm no longer is there once it shines. Okay. So now the next question was, so how? I mean, we have found serotonin, we found this neuropeptide, we found this receptor, but how do they come together? Why do they cause a swarm formation? So Navneet, when she was thinking about it, she thought maybe what is happening is these worms can no longer touch each other properly. That is, they are no longer able to recognize touching of each other. Okay, so that was her thought, and the reason she thought this was previous work has shown that this PDFR1 receptor is expressed in mechanosensory neurons. People are not actually seeing whether it affected mechanosensory neurons or not. So the first thing she did was she took a mechanosensory neuron, actually she looked at multiple neurons that were defective in touch sensation in worms, and she found that largely they did form swarm formations. So then she thought, okay, if this is the case, can I actually just touch them on the head and see whether they're able to recognize the touch? And what she found was in the anterior region at least, Cas1 mutants and PDFR1 mutants are unable to look at touch properly. They look like the mechanosensory mutant, which is a mechanosensory. In the both in two types of anterior touch, this is the case. However, in the posterior region, everybody is fine. Only the mechanosensory mutation shows a phenotype. And this is not surprising because whatever we've shown with Cas1 is in the anterior region. Cas1 is not even expressed in the posterior. So it seems that the swarm formation may be to some extent because the worms are not able to sense each other properly and hence they come together and stick together. The final question that we asked along with our collaborator was, is there any relevance to this behavior? Does this give any advantage or disadvantage to the worm? And for this, our collaborator essentially took Cas1 mutants and wild type worms and recorded them on a lawn of bacteria over a period of 3 days. So they looked at multiple generations of worms to see what was happening in various situations. So this is Cas1 mutant and below is wild type. You see, wild type just moves everywhere. Cas1 mutants largely stay in one side, that is towards the right. Okay, and this is just over generation. Wild type is moving everywhere, but Cas1 stays on one side and it swarms. So clearly these mutants are essentially sticking to one side and they're forming a swarm as they go. Now we think this might actually not be advantages, this might actually be disadvantages because now some of the worms are not going to get enough food, the worms that are in the middle of the swarm are not going to get enough food, Charmaine, who's an Erasmus Mundus MSc student who's currently in Germany, and Dimple, who's joined Biocon after doing a master's thesis project in our lab, and Emil Saratikin and Aslim Okavas from Turkey for helped us with the last experiment that I showed. For environment, gave us a lot of strains and optogenetic reagents. Arnav Mukhopadhyay gave us a strain. Raghav showed me that paper where the science paper which we looked at and saw that maybe serotonin is involved. All the members of our lab, and I especially want very grateful to Center for Genetics, Serotype and Genetic Center, CTC, for giving us work. India, people works, biorecords, what Naveen used to make all the graphs, and all the illustrations. And I'm very grateful to NRF, India Alliance, Department of Biotechnology, and ISC for funding. Thank you.
Ma'am, we sincerely appreciate your time and thought-provoking insights you shared in today's session about Cas1 neurotransmitters and neuropeptides and about serotonin. Thoughtful questions and explaining them very clearly. Thank you, ma'am, once again.
Thank you, madam, for your wonderful lecture. It was both informative and inspiring. We truly appreciate your time and the knowledge you shared with us today. Thank you, madam. If anyone of you has any queries regarding to this session, you can ask. You can kindly come forward and clarify your doubts or queries. I repeat it again. If any one of you have any doubts or queries regarding to this session, you can kindly come forward to clarify your doubts.
Any questions from anyone?
Yeah, you can come forward to ask the question.
What are the major challenges? Okay, so these are not easy experiments, unfortunately, to do. So when Navneet did these experiments, she had to stage the worms. They had to be perfectly staged. She had to have this correct thickness of bacteria. We think, and these are something that the next PhD student is looking at, we think that maybe if the bacteria does not have the correct set of metabolomics, like the metabolites are not correct. Sorry.
So now it's on now. Now you can hear me. So we think that if the bacteria does not have the right set of metabolites, we may not see this, we may not see what's happening. So she had to go through a lot of iterations of making sure that the bacterial log was exactly the right protein, that the bacteria was the right protein, that log was the right size. All of that she had to go through multiple iterations. That was the main challenge, and then for the optogenetics experiments, it took a lot of troubleshooting to make sure that how long are you going to put the blue light, how long are you going to put the red light. All of that took some figuring out. But other than that, it was essentially she was very motivated to do it, so it was not. I don't think it was that difficult.
Any more questions from anyone? You can come forward and clarify your doubts and queries.
No others?
Thank you for the wonderful session, ma'am. Actually, in the first slides you have connected the human brain and the cell areas. And actually from this study, how do you extrapolate this behavior in humans?
Okay, so we can extrapolate. Again, this is going to be eyewash. There is no real actual data. But so a lot of work has shown that calcitogens are required in autism. Okay, in autism, you do know that touch sensation sometimes shows up in autistic patients. where they're not very happy being touched or with people around them. So maybe one can imagine that maybe this could affect, could be because of the touch sensation. Now what we are doing and what graduate student Siddhant is doing is, he is humanizing these worms. So he's essentially putting in this complete Cassivan widget, he's putting back the human Cassivans one at a time to see if they can rescue this phenotype. Now if they can rescue this phenotype, then we're thinking we can use cells to see who Cassivan, the human Cassivan, interacts with, and then see if there's a counterpart in C. elegans to see whether those interactions are actually real in C. elegans.
That was wonderful.
But one of the things is that unless you have a baseline and you have something to study, you really can't do much. You can't go, decide to put the human counterpart.
Any more questions? Thank you, ma'am. Thanks for answering all the questions. Thank you, ma'am.
Now I request the co-chairperson, Dr. Raghuveer, Indo-American College of Pharmacy, Jakarta, Nasrapet, to address the closing session. I request Sir Dr. Raghuveer to kindly come on to the desk.
Give a big round of applause for sir.
That was actually a wonderful lecture, ma'am, Dr. Walker. And for the students' point of view, see, there are a lot of questions which were asked in their lab, and that brought this beautiful work. So, for science or research, curiosity is the key. So, in the labs, every time we have to question ourselves, question our hypothesis, and then design experiments. And that was actually, when I sat there, I was actually having goosebumps. So the questions you asked and the way you done the experiments and all, that was really, really good. And to summarize what Madam has delivered, so we learned how the simple model organism C. elegans helps us understand complex brain functions, and the role of Cas1 gene in regulating serotonin, and also the receptors PDR1 receptors in the neurons, how they were related to the smart behavior. So all these actually the questions they have put in their lab, and how they have answered and generating mutants, and someone has asked actually what is the difficult task. Actually the entire thing is difficult. Because someone who has a biotechnology background would understand how difficult it is to generate these mutants and all. So, because fortunately I had an opportunity to be in a biotechnology lab at ICGP New Delhi with Dr. Ranjananda. So I know how painful these experiments are, and really great thanks to your student Navneet for bringing out all these experiments. And with this short note, I congratulate and I appreciate the speaker, today's speaker, Dr. Kavita Babu, for this wonderful session. Thank you, thank you.
Thank you, sir. Thank you so much for addressing the closing session. Now I request all the dignitaries, the advisor sir, the global secretary, the principal sir, and all the faculty members and all the other speakers to kindly come on to the dais to felicitate Dr. Kavita Babu ma'am and Chair and Co-Chairperson
సార్ క్లోజ్ అవ్వరా కొంచెం బ్యాక్ జరగండి సార్ సార్ కొంచెం ఓకే
It is our great pleasure to felicitate Dr. Kavita Raju ma'am, and now it is our privilege to felicitate Chairperson Dr. J.V.S. Suresh Babu, and request all the dignitaries to felicitate the Chairperson.
బ్యాక్గ్రౌండ్ లైట్ వల్ల రెట్ల కావాలంటారు అది ఆప్షన్
సార్ కొంచెం బ్యాక్ సార్ రెడీ సార్ చూడండి
I request all the dignitaries to felicitate Dr. Kavita, Chairperson Dr. Kavita.
సార్
Thank you, ma'am. Thank you, sir. Now get ready to witness grace, rhythm, and boundless energy before we continue with our next scientific session. Let's light up the stage with an electrifying dance performance. A big round of applause as we welcome our talented AMR students.
तो भी आगे वाले क्या करना नहीं जब लगा स्टेज पे कर जाने वहाँ
जरूर डाल लेंगे
कालू सामने बहुत ना बोलते हैं सर चल गुड लॉन्ग रख लॉन्ग रख सर
provide a glimpse into his outstanding and professional achievements before leading the session.
Give a big round of applause for welcoming our chairperson and co-chairperson onto the dais.
Respected chief guest, esteemed faculty members, dear students, ladies and gentlemen, good morning to everyone. It is my great privilege and honor to invite a distinguished academician, researcher and administrator, Dr. Pamureddy Bhavan, to the stage. Dr. Pamureddy Bhavan holds a PhD in pharmaceutics from Andhra University, Visakhapatnam, along with master's degree in pharmaceutics and bachelor's degree in pharmacy. He has more than 18 years of rich teaching, research and administrative experience in pharmaceutical education. Currently, he is serving as a professor and a principal at Nirmala College of Pharmacy, Atmapur, Mangalagiri, where he has been contributing significantly to academic excellence and institutional development. Dr. Bhavan is recognized research supervisor, having successfully guided PhD scholars, M.Pharm, B.Pharm and PharmD students. His academic contribution included 22 research publications, 1 granted patent, 1 published patent, 5 chapters and 2 textbooks. Due to his active participation in several faculty development programs and his active outstanding contributions to pharmacy education and research have earned him prestigious recognition including Dr. APJ Abdul Kalam Lifetime Achievement Nation Award and Best Researcher Award. He has also received research grants from AICTE and has organized several national level academic conferences demonstrating his commitment to advancing pharmaceutical sciences and research. Ladies and gentlemen, please join me in the extending a warm and enthusiastic welcome to Dr. Pamureddy Bhavan. We are truly honored by his gracious presence and look forward to his valuable thoughts and inspiring address. I now respectfully invite Dr. Bhavan to kindly grace the stage and take the chair at present for today's session. A big round of applause. Thank you very much.
Thank you, sir. Very good afternoon to one another. So it is a great privilege for me to introduce the Dr. K. Krishna Chaitanya Foundation. So Sir has completed his MBBS from Siddhartha Medical College, Vijayawada. Sir has completed MSc in Orthopedics from the NRI Academy of Sciences and Senior Resident from the Government General Hospital, Vijayawada. His leadership and professional positions. So Sir has served as a Program Director, FMB Spine Secretary, and Executive Committee Member, ASSI, and Fellowship Committee Member, APSS, and member of Spine AP, Cross APSS, ASSI. And Sir has received some awards and honors. Sir has received the Spine Asia Pacific Research Grant, and also Sir has received the Indo-American Spine Heritage Fellowship and ASSI Traveling Fellowship. And also Sir has received the prestigious Dr. Dharmandri Medal. And Sir also has been awarded some multiple national and international presentation awards. Sir also has been continuously in the research and academic contributions. His major area of expertise minimally invasive spine surgery and robotic spine surgery. At present, Sir is working as a Head Department of Spine Surgery, Program Director, FMB Spine Surgery, Krishna Institute of Medical Sciences, Hyderabad. These few regulatory points, I request everyone to put hands together. I invite the today's speaker. Thank you, sir.
serious speakers. There's a wonderful research, including the blood bank, in the brain bank, and also the biotechnological research which is going on. All these are the background for what I actually do now. Because my work, which I'm actually doing now, is just 10%. The 90% effort is given by various researchers which have been going on, which are given to you by the presenters. And it's amazing to see such good, eminent people coming all the way to Narsopet to educate you children. The most important thing, I only feel that they are here just to inspire you, so that at least some of you will be interested in this research and will take our country forward. So, in a way, I need to say that India is good among everything. Population is our strength, but still we are lagging behind in multiple aspects of medical sciences. And the first thing I tell is research, because we are all acquiring technology from various countries and using it, but we still are not among the first or second in enabling those technologies, so that we would continue, we would make them in our country, which is much affordable, will be much cheaper, and also much beneficial for most of them. Okay, so these are the efforts made by your institution management, and I appreciate that. And we are also together with them to inspire you guys. So I'll make my presentation very brief. I just want to show you how robotics in spine surgery work, and I'll show you some patients which we operated. And I'd also like to tell you that there is only one spine robot in AP and Telangana. There are hardly 3 to 4 in our country, and the one which we are using now is very advanced in the entire world. So we are first to acquire this in the entire world. Nobody else has it, and now recently a couple of other institutions have acquired it, and we are doing great numbers. Coming to the human spine. So, whenever you think of human spine, it is the backbone of your life. It is something. Yeah. So it is something which is used to support your body. So the entire weight from your head to toe transfers to the spinal cord. As we know, we have divided animals into vertebrates and invertebrates before, because vertebral column is very important. Apart from transmitting the weight through your body, the other important aspect of the vertebral column is to be flexible enough and also to support your spinal cord. Okay? The spinal cord, as all of you know, is something which carries the entire information from the brain, neurological information from the brain to all parts of your body. Okay? So this is the spinal cord, and this carries the entire information from the brain, right? So it's divided into cervical, in the chest area it's called thoracic, in the lower back it's called lumbar, and later it's called as coccyx and sacrum. Also, it is made up of 33 vertebrae, and there are some discs in between that which make it little flexible. So, what are the spinal problems we commonly come across? So, most of us tell that I have a back pain, I have a disc bulge. So, to make you understand, the most common problem is a slip disc, where the intervertebral disc, which is in between the two vertebral bodies, whose purpose is to make it flexible, sometimes get degenerated and slips and goes and hits one of your nerves. This is called a slip disc. So when there is a slip disc, nothing to worry, but you come to a doctor, telling I have a lot of back pain. When I am sitting for hours together, maybe 1 or 2 hours, I have severe back pain, I need to lie down, again to sit back again, I need to lie down for 30 minutes if you want to do. This is the most thing which you come across when you have a slip disc. Next thing is sciatica. If this disc is even bigger and involving one of your nerves which is going into your legs, probably in your low back, that is called a sciatica. Then you have a severe leg pain. It is a more difficult and a more serious consequence of a slip disc. The other one is a fracture, when somebody comes across an accident and then they break their bone, and it might hit sometimes your spinal cord as a fracture. So these are the common things which we come across. The rare things are some tumors, because spinal tumors are the most common tumors in the entire body, because whenever there is a tumor in lung, brain or anywhere, the common area it goes to your spine. So it spreads commonly to your spine. The other one is deformities, which I am going to show you some patients, where I am expert in, my expertise lies in, where some children from birth will be bent. So if you don't look straight and you are bent to some extent, it is called that you have a scoliosis or a deformity. So I will show you those things. So these are the common spinal problems. As I told you, that is the slip disc. This is the spinal canal stenosis, where you can see the spinal cord is diameter is very less. This is a fracture where you can see the vertebral body is lost its shape because of injury or any fall. This is scoliosis. then you have to be straight your NTM, which is scoliosis. And there are multiple tumors or infections can happen, like tuberculosis, bacterial infections or tumors, anything can happen. So, do you really operate everyone? If some problem comes in spine, is surgery the only option? Definitely not. 99% of the time, all these problems can be treated without any surgery. It can be treated with good physiotherapy, good rest, because in spine what we do is only a damage control. That means you just control the damage. You just cannot prevent it. But our body's excellent repair mechanisms are present. Even a slip disc with 80% of spinal cord injury can recover on its own without any treatment. So our body is prepared in such a way that all these problems can heal on its own. The doctor intervention is only required when you feel that the time required for healing is way more and they find the neurological injury which can happen before the disease. Only at that point of time, the doctor will intervene. So, coming to my topics, what are robots in spine? Have anybody of you seen a real robot working outside anywhere? Like you've seen any car or you can see the car manufacturing companies doing many videos of robots picking up the parts and assembling together. Similarly, robot is nothing but a device. It doesn't look like a human, or it doesn't look like a man. It's just a device which is used to perform activities which are ordered by some human being. That's it. It looks like whatever shape it's required. It doesn't look like this as such. And coming to the robotics in spine, the actual who performs the surgery. It is not the robot which goes and performs the surgery. It is the surgeon only who performs the surgery, but with the guidance of the robot. Why do you actually need robotics anywhere, in any field? Because human beings have an error called as human error. Because you want to do some activity, you can have a little bit of imbalance in the precision, you can have a little bit of calculation error, you cannot judge the angles perfectly. But if you assign this to a robot under your supervision, you can tell it, go to 30 degrees angle in AP view and put it in a 7 mm quarter angle. So that is the perfect. With your eye, you might not have to do it, but robot will do it. But under your guidance, you have to prompt it to do. It doesn't do anything on its own. Okay. So this is not which happens in the operation theatre. Robot doesn't perform the surgery. It is the surgeon who always is in control. So coming to surgeon versus robot. See, basically, once we acquired a robot in Hyderabad, which is the first robot. So most of the students who come to me think that, okay, once the robot is there, we need not learn anything. We can just do things in the computer and just do an operating machine. Absolutely wrong. Because when you do something, you know what you are doing. But when you need to control something, you need to be more cautious. That means controlling someone or correcting somebody's work is more difficult than doing it. You have to be absolutely precise. Even a small lag, a small misinformation related to the robot can cause catastrophic complications. You don't have a second chance when you are operating children of deformities of 12 and 13 years of age. You just cannot explain the complications to them and the results will be very risky. So, robot doesn't replace the surgeon. Always surgeons or human beings are in complete control of whatever we are doing. So this is a kind of robot which is semi-autonomous, which we use in abdominal surgeries or renal surgeries, where the surgeon sits in a console while the robot operates. We don't really control them, but we control them from a distance. So this technology can be used to control surgeries even from other countries. So I will be sitting in Hyderabad and I can control the robot by internet operating in Vijayawada. This we can do, but this semi-autonomous, even here, the robot moves only with my directions, it doesn't move on its own, but of course, sometimes there might be lag in the transmission and there might be error, but that will be corrected with further technologies. But in spine, we don't use it because spine is too risky for such semi-autonomous things yet to control. So how does it work? If you want to operate a patient with spinal problems using a robot, first we take a CT scan. You all use Google Maps like this. Okay? When you are traveling in a car, from here to Hyderabad, from here to Visakhapatnam, whatever, you use Google Map. What the map actually does, it takes the, you have a map of all the roads and everything lying between Vijayawada to Vizag. So, on that, your car will be superimposed by using GPS. In the same way, the patient is mapped by doing a CT scan inside the theater. So you know what parts exactly are there in the patient. So all that, my arm will be superimposed. So my arm and the instrument which I am using. Along with that, it will also guide us. It is like a car working on a Google map with automatic driving. We just need to sit and observe. It's a little more advanced than this map, but it is also having an automatic driving. So that is what you do. It is like a navigation during surgery. So this is the workflow where we have a CT scan inside the operation theater. You can see from the CT scan you get images, and these images when I am operating, the screw is or something which whatever I am doing on the surgery will be superimposed on the pre-acquired CT scans. So once the CT scan comes, you plan everything on a computer. You don't touch the patient, you just stand in front of the computer, you plan, okay, I need to do so and so things and so and so cases. And then, once this is done, you can see my arm is having these trackers that I am operating. And in the screen, you can see exactly where I am doing. So I did not open the patient, I do it right from the screen. So I can see whatever is inside the patient and I am operating from outside. You can see the robotic arm is guiding me, and my arm and the instruments all have these lions and trackers. So exactly in the screen I can see where I am putting the screw, where the vertebra is, where the canal is, everything can be seen from outside. So why choose a robot? So if you ask me, I have been operating from last 12 years in this hospital. I have done more than 10,000 surgeries. Even before this, I was doing my fellowship time. I was also done some around 4,000 to 5,000 surgeries. But even then, there might be an error because I do without a robot in 1,000 patients. If I put some 10,000 screws or some cages, one or two might be wrong. But with the robot, even that will not be wrong. Because you know where you are, and also you can recheck immediately after the surgery by doing a scan again to confirm that everything else is normal. So, in human life, you cannot expect 1% complication. If I operate around 100 folios a year to take. If I create paralysis or loss of power, sensation, or completely paralytic like two children, that is also considered a complication. Even that cannot be accepted. So you need 100% precision in everybody. Nobody wants to be having any complication. This is unacceptable in surgeries in spine. So we need 100% precision. That is the reason we went for the robotics, where we yet hadn't had any shortage or any complication yet. We finished around 200 surgeries in this robot, and we hope many other institutions in India also will require this. And so we wanted to so we have greater precision, less blood loss. It's a minimally invasive kind of thing. Simple like when you are lost in a forest, if you have a gun, you can come out easily. That is the way I can explain this. So is robotic surgery better? Definitely better, because correct diagnosis, skilled surgeon, proper patient selection, along with robotic technology is always better. Surgeon is most important. You need to operate on a patient only when it is required. You need to be very loyal. And my professor, Dr. Rajshekar, I was in Ganga Hospital, the best in the world right now, I would say, was telling only one thing: you need to do self-testing. If you are in the situation, if you are operating the patient every day before going for the surgery, you have to think once and yourself, will you do the same thing if your mother, father, or your loved ones is in the same situation? Yes. If you pass the test, you go ahead. If you don't pass, you wait, think, and see what else is better. If you feel some other surgery is better, some other technology is better, you better refer to that. So this is what we practice every day in our hospital. The self-testing before you go. Once you do that and you follow these things, I think your results also will be good and the patient also will be much happy. So if you ask me how robotics evolved, so this is most important thing. Intraoperative localization of surgical instruments. This started way back in 1980s in USA. Every technology which we acquire now is from Europe and USA. But soon we will try to change this with the help of Internet professors in front of us and their students also. So the results of this surgery indicate that computer-assisted techniques significantly improve the accuracy. So these are the images of first intraoperative localization. That's what is neural navigation, which was started way back in 1984. This is the original article. And then comes spine assist in 2004. This is the primitive robot. You can see this doesn't have any navigation. It just moves on to the particular pitch you want. It stays there. And from the small hole, you try to put it. This is the first robot in 2004. The final robot which was created is not in India. As such, it was only in other countries. So then comes the Lizard Renaissance in 2007. It is the second version, where it is a little advanced. The only advantage is it has a small bar which can actually prevent skidding. I don't want to go into details, but it's more precise than the original one. Next is the Mazon X. It started in 2016, and this is what we have right now. It is a newer version of Mazon X. It looks like this. It's more advanced, and you have something to, you can see what the road is doing in Mazon X by using computer navigation in the 3D display. So the other road is Global Sensor CS, both are the same in a different company. So definitely precision. So in medicine, everything is evidence-based. Everything is scientific research oriented. So I cannot say that I use a robot and it is better. I have to go back to the literature and see what has been done till now, and is it really good or not. Or else if I want to change something, I should do my own research and then present it to publish. So I haven't done it, but we are in the process of doing. But all the papers in all the review articles which have been given before have shown that definitely robotics is better in most of the cases. And regarding accuracy of it, better. But there are few papers which suggest that there is no difference, but very few, one or two, but most of the articles suggest that robotics is much better compared to I will show some cases. This is 20-year-old child. You see that she is bent in the back. You can see X-ray. She has a severe deformity, and it's around 60 to 70 degrees curve. So the idea is, in India we don't have school screening programs also. We started right now in most of the states with ASSI collaboration. That means that every school should screen the children at 5 years and 11 years of age. You have to routinely screen all the children for school ages. Otherwise, these deformities will become very bad in future once they reach 12, 13 years, and that will cause a problem for us thoracic inflammation to see. Where your lung will start developing at 3 to 4 years of age and reaches 80% of its maturity by 7 to 8. So if there is a big curve in your chest, physically it will never allow your lung to grow to its normal capacity. Then you start a disease called as restrictive lung disease, which can be so worse that children might not be able to survive up to 30 to 35 years of age. So to prevent that, you need to identify these causes at an early age, and you need to prevent it from worsening. I identified one child where we identified at 6 years of age, but since you cannot operate at that age, you will try to put some rod and try to expand that rod for every 6 months. Once she becomes better, like around 13, 14, then she gets 80% of the lung capacity, and she develops most of her spine is crossed, then you start operating on it. So in this child, we did a robotic planning. These are the robotic screws which are flat, and it is a post-operative surgery where we corrected the entire spine using screws and all. So if you see this planning, there is a spinal cord inside the bone, and also there are multiple vessels like aorta, vena cava, lung, heart, everything adjacent within 1 to 2 millimeter distance of the spine. So any screw breach or any injury to anything, you might end up sometimes even in a death operation. So very difficult. So you cannot go mistake something you are putting a nail in a wall and you have to put it in a 3 millimeter area. You have to put it in a 3 millimeter. You cannot have an error here. That is the reason we want to use robotics for precise. So this is the X-ray of this. So I will give you one video where this is how the robot is mounted at operation theatre. So I will run this video since it's not a scientific session. So this is how the robot automatically scans and sees where different parts of the child are present. So you will check the accuracy of the robot by putting manually on different parts. Once it matches to your anatomical landmarks, then you ask the robot to start operating. So this is how you check the accuracy. So once the films are on, once the CT scan is done, then you start planning the screws on the console itself. Like that, you plan the screws on the computer. Once the planning is done, you just ask the robot to move to right side towards the right radius. And exactly goes onto the trajectory and sits there, and you can put a screw from the center of the trajectory. how it goes to the specific trajectory. And from that, start operating. So once you put all the screws, then you remove and you try to correct the whatever by using smart links. You have various devices. And you also try to decompress this final part by going, it's got a more scalpel. It's also one of the most advanced technologies where it is an ultrasonic bar kind of thing, where you cannot, it only cuts the bone. Once you go and touch the blood vessel or the spinal cord, it automatically stops. For example, if I give you a scalpel and ask to damage my hand, it is impossible. You just cannot do any scar or something. So that's a very good technology. So we use all these latest technologies to operate on such patients. This is how we derotate the spine at the end. And we use multiple tools and rods for that. So then we also verify to the CT scan in the operating theatre itself. So then you see all the integrity of all the screws, you check everything very clearly and you confirm. Along with that, we also use intraoperative neural monitoring, which I show you in one of the other patients. This is the final surgery. So that is the outcome. So this is another child. See, this is most important because this child came to me at 7 years of age with this deformity. So it started deteriorating, but I put her on a small brace, and when I see her, within another 4 months she became very big. So the child is only 7 years. So if I do the surgery which I showed you in the before child, she will not grow after this. Because I put screws and fix her and the height will be the same, her lungs will not grow. So we had an innovative technique which is called as growth rods. Where I put small rods inside and expand them every 6 months. Every 6 months I do a small surgery and expand them till she becomes 30 years or 20 years of age. So that I will preserve her growth and also preserve her lung and heart function. So this is what is called as growing rod. You can see the small joint in the middle. So that can be expanded every 6 months. So this is a 2022 and you are 8 years of age. And after 2 expansions, this is the situation. And after 6 expansions, this is the situation. And now in 2026, this is the situation. The important thing is that is to convince the child to come close every 6 months. In India, the problem is follow-up is very difficult. You do a small surgery on somebody and you never know when they will come back to us again. So you have to stick with the parents. You have to explain them. I think that this is the important. If you don't do this, you will have. I showed one child who had a congenital student who came to me at 7 years old. Didn't come back to me again. And I just show you what happened to her. So this now she is around 12 years and now she is ready for a final surgery. So then this is the robotic setup you can see. The table which we use also is important, and this doesn't have anything which is metal, because if there is a metal, the CT scan will not be allowed. We cannot do a CT scan on that. So then we do a CT scan like this, and then we clamp the screws everywhere, and then we expose the patient like this, and then we put the screws. You can see I am putting the screws to the robot, and I can see exactly where the screw is going. If the patient or anybody is trained, it is easy to do something. But if they are completely twisted in 3-dimensional direction, and you don't have error of 1 millimeter, you cannot accept 1 millimeter error. If I don't imagine the screws in a proper direction, and if I misplace them, it is very risky for the child. So this is okay, but you can see the other video where the spine is like this, or the vertebra are twisted. So I had put the screws parallel to the ground. That means when the spine is like this, I have to put the screw like this. So this angle you will never get without a robot. So that is the importance of the robot. You can see they are putting the screws almost parallel to the ground. Very normally it's very difficult. Almost on the skin and it's going into the whatever. So that angles, whatever you know, whatever you need at the time of surgery, the robot will help. Otherwise, you will have to go and take an X-ray and then see the spinal cord, go and take an X-ray. It will take 2 to 3 hours just for putting one or two screws in this. So most of the times you need to operate for 18 hours. Again you close the child, come back after 2 days, again you do the surgery. Again you go, again come back from there. So it will take 4 to 5 sittings for operating this. But now this technology we can finish off in a single sitting under 5 to 6 hours. So you can see we are putting the screws. Exactly see where it is going. You can see the center hole in the spinal cord very nice. Here you can see that last 20 seconds you can do. Video and fast forward. No. Here. Next week. This one to forward. You can see the angle here. It is almost going to the skin and the vertebrae is totally rotated. So that is the importance of this also where you cannot always get the angle you normally put from the above, but here you are doing from see the vertebra how it is rotated instead of being straight. So that is the importance of robotics. So then you do this derotation maneuvers using smart links where you try to track the wings and also turn the spine. And this is the post-operative age of the child. You can see this is a very good touch. So this is where she came to us. This is the course of the disease when she was 7 years. And now this is 12 years, it is a permanent fixation. She will not deteriorate from here. If we don't identify them at 8 years, this will be the situation. So this child is now 13 years. I saw her at 8 years of age with a small deformity. She didn't have the X-ray. And you can see her lung is collapsed, her heart is collapsed, she is breathing, gasping. We did the surgery for 5 days. Just imagine it more than 100 hours sometimes in the middle with a gap. And did 4 osteotomies to 50% correct that. But I would tell this is much better. So I don't want people to come to me at that stage because it's very high risk. The child has to stay in the operation, they have to come in ICU for 2 months after the surgery. Now he is fine, but definitely this is very good. So this is another child who came to me with a congenital deformity, who was walking till 13 years. His parents were somewhere from over here or nearby. They didn't have the effect of getting him at an earlier stage. They thought that was a minor deformity. But once he stopped walking, once he developed paralysis in the legs, he was unable to stand and walk, and they came to me. He was unable to stand in the situation when he came to me. You can see the X-ray of this child, and you can see the MRI. You can see the spinal cord is completely compressed. There is no way you can get out of this. And is already paralyzed. As you seen from our previous speakers and emerging, we just know tip of the iceberg in here. There is no medicine or there is nothing in this world which can recover a damaged neuron. Once a neuron is gone, it is gone. You can never give a medicine, you can never do anything for it to recover. If it recovers, it is in a neuropraxia stage, it will recover on its own. Otherwise it will not recover. You have nothing to do. You just can remove the compression over it and wait for things to happen. That's it. Once it is gone, it is gone. So this child came to me in this stage that he still has some power, some sensation, some moments in the leg, which are good. So for these children, we have a technique called as halo-therapy traction, which is not done in India so far. This is the first case which we did. We put a halo on the head and try to stretch the deformity by using the weight. You can see the weight there. So this has to be put on the child day and night for 2 months. So the child stays like that even in the night for 2 months. The idea is to stretch the deformity for some extent. The major idea is to accumulate the spinal cord for traction. The spinal cord will be stretched in this time so that it will be used to corrective activities. Otherwise, when you at the time of surgery, when you slightly try to move it, there will be permanent damage to the spinal cord. You might correct the deformity, but the child will not walk. So we didn't have for 30 days all this feature and all the makeshift arrangement because in the US there is a halo traction available, but in India we don't have. So we have to wear all these things and create a halo. This is the neural modeling signals which we tried. You can see that means while operating, what we do is pass current from the head and try to record on the feet. It's very simple. Here we are not getting any signals. I am stimulating the child. He is moving his hands, but he is not able to move his legs. That means the signals are not coming down. That means the spinal cord is so sick that we are not able to get the signal. He explained the same to the parent also, but we had to go ahead with the surgery. So we did a vertebral column resection. That means we removed 2 vertebrae in the bone and we reconnected the spine again. So this is is how we did this surgery. From 90 degrees to 50 degrees. 30 is normal, but we could not get into 30 degrees vertical. So this is after the surgery, post-operative. You can see he's walking a little bit. This was 4 months before. He's unable to put his leg, but a little bit of movement is there. He's trying to walk. It's very good because when he came, even this was not possible. And this is now when he started to walk. Okay. So this is happened. So it is never there, but definitely you need to explain them. See, doctors are under a lot of stress because when we get 100 patients, we cannot expect 100 good results. There will be some failures. But the family needs to understand that they are not God, they are human beings. But we try our best in every possible case. I haven't seen a doctor being negligent except very, very rare occasion because everybody will try to do their best. Because at the end of the day, is another human being, so we will try to do whatever is best for the child. Okay? So this is another child who came to me 2 years of age with a small throat infection. Throat infection can cause injury to the cranial junction. That means very rarely it can cause infection at the spine also. So you can see because of infection here, she lost one of her most important vertebral bodies, which is called as C2. So whenever she tried to move her head, her neck and head are moving independently. It's called a atlantoaxial dislocation. It's very risky because child of 2 years you cannot ask her to sit quiet with a hammer, with a collar and all, and she might land up in respiratory paralysis, as Madam told. Medulla is the most important region of the brain. In her top, where most of the important neurons are present, in mitochondrial regions are also not present there because if there is an injury to the medulla, you cannot expect respiratory paralysis. So the most important region of the brain. Forget any other region, the most important. So you can see we didn't have an option other than putting the child on this kind of brace for 3 months because children will definitely run and do anything. So sudden movement can cause injury. So we tried without a surgery, but we imported this hammer from another country when this is not available here. So we put her in a hammer after 3 months. Even after 3 months, she did not improve. So we went ahead and did the surgery using a robot in this case also. So the bone in size was only 2.4 and we had the lowest screw available in India was only 2.3. So we had to put that screw and there was only one attempt to put it. So luckily it could place the screws using the robot and finally we didn't have a bone to put on the screw so that it will use to remove it graft from her mother and we placed it. So we did a surgery there and now she is almost 4 years after the surgery and she is perfectly fine. She is doing. So these are some things which we see. And if you want to see some robotic steps, I actually told you I will skip this video. So what are the myths versus what are the facts in robotics? The robot performing surgery by itself is completely wrong. The surgeon is in control in every step. It is experimental. Johnson protocol, as I told you, it started in 1984 and now it's third generation robot. And it is the surgery which is used worldwide. And in US Michigan University in 2016, we were using that robot. In India, it is in the some late. It's not for complicated surgery. Simple surgeries, complicated surgeries. We don't want failures in any complicated surgeries. Patient might accept the basic or simple, but more failures because nobody will accept the failure. So we need robot everywhere. It is extremely safe. It's FDA approved. It designs with multiple checkpoints, but the most important thing is the surgeon should have enough experience. Surgeon should be experienced with the normal surgeries for at least 5 to 10 years before going on the robot. Otherwise, it's going to be very risky. And the future of spine surgery, artificial intelligence, and you can add an artificial intelligence where the robot might really operate in future without the surgeon or surgeon sitting somewhere in other countries also might do the surgery. Maybe it is possible. It doesn't make, robot doesn't make our life easy, but it makes our life better. Okay? If you don't think that once you get this technology, it will be very easy for you. More risk. As you know, the privilege comes with more burden. You cannot expect life to be easy. You have to be more conscious with the technology. Patient or anybody, it doesn't matter whatever you do, they want the result. You can use a robot, you can use another tool, you can use another tool, but it is for your comfort, but not for the patient's comfort. You are using this technology. Okay? The best surgery combines advanced technology with the experience and judgment of a skilled surgeon. Okay? And this is my colleagues in Malaysia and Calcutta and Singapore, there I have worked before. Thank you very much. Thanks to Dr. Bhaskar for giving this technology into India. It is not easy to acquire this technology. Very expensive. And somebody like you to trust me and get this technology to be the first in the country to operate this technology is amazing. And also, you would expect all the students here to get inspired by this and choose your choose your path and start working for that. Don't expect the results, just do whatever you will do. Automatically, you will fall in the right place to have the most. Thank you very much.
Thank you, sir, for sharing your expertise and inspiring us with your knowledge about robotics in spine and spine surgery and showing us your inspiring work. Let's give our esteemed speaker another big round of applause.
Thank you, sir. Thank you for giving a wonderful lecture. And students, it is now your opportunity. So grab it and come forward to ask any queries or doubts regarding to the session given by Dr. Chaitanya, sir. It is a great opportunity to interact with sir now. You can kindly come forward.
Can robotic systems adapt in real time if your patient's anatomy changes during surgery?
Yes, that's the most important of robot. You don't prepare the robot before surgery. In the time of surgery, you do a CT and all the anatomical anomalies will be taken into account before the robot installs, that means before the robot gets ready for the surgery. So, in normal patients, a surgeon can do, but in anomalous vertebra, we cannot identify. That is the most important thing of the robot. So, robot will be different for you, different for me. Your CT scan will be taken by the robot, not my CT scan. So, your CT scan will show the anatomy. If it is one vertebra is absent or one vertebra doesn't have a vertebra, you cannot plan the screw there. So, definitely that is the most important thing in robotics. It will consider all your anomalies before going on to the surgery. Without the surgeon, it can automatically do the surgery. Okay. So there are certain robots in abdomen and knee which will work without the surgeon being present. But what happens if you operate on a knee? A small error might cause a little bit deviation in the implant. In the abdomen also there might be a perforation in any of the abdominal organs and it can be repaired. But in spine, it is very risky. That means if there is an injury to the spinal nerve or a root, we cannot repair it. There will be complete paralysis to the cell. So right now there is an artificial intelligence integrated to it, but we are not using it because we didn't get the approval from the FDA yet. Because even a little bit of error cannot be accepted. So it will take time for further refinement, and definitely in future, what I can do, I can just put the X-ray of the child in that, and the robot automatically plans the screw. It will automatically tell you what you have to place. It will automatically put the screws in, and it might also derotate, and you might have to just check it. But it takes time, right? If normal robots in other departments takes 5 years, in spine it will take 20 to 30 years. Because the error is unacceptable in this case. So it has to be completely precise. So the refinement takes. So it will take time, but definitely it will come in the future, hopefully.
What are the technological and regulatory challenges majorly?
So major regulatory challenges challenges are there are multiple steps which are involved in allowing the robot to come to the market in spine. Because knee joint replacement robots, every hospital in Hyderabad has it. But if you take for spine, there is only one robot in the entire every country. That's not it. So if you need to acquire technology, there are multiple these things from the government. So we have to show that it is safe. We have to show that the surgeon is trained here. There is a long training session. There is a long experience required. And I have done so many deformities. I know how to do a deformity, and the robot, I will see that it doesn't go wrong. So that is something also. If somebody comes to us and it's a new technology, you tell them, sir, if it does a mistake, what will you do? You can say that I am there. So it's just assisting me, but it's not doing any surgery. Because in some conditions like brain and spine surgery, the risk is very high. So there you have to have so many guidelines and regulations. So it's important to the government as you asked me, and also the cost is another huge burden because it's very, very expensive. So it's very difficult to acquire this technology. So these two are the things which I think are the most difficult things which we should cross before knowledge.
Thank you, sir. Thank you for this information.
Good afternoon, sir. How does robotic surgery can improve patient outcomes when compared to traditional surgery?
In robotic surgeries, as I showed you, before traditional surgeries, precision will be coming with an expense of time. So if I need to put one screw in a patient with such a big deformity, I will take 2 to 3 hours because if I rush it, I might have an error. But with robot, the time will come down because you can do it in 15 to 20 minutes per one screw. So the surgery which usually takes for 3 to 4 sessions and a couple of days long will be finished in 5 to 6 hours. One is the time. Because once you reduce the time, the anesthesia will reduce, the blood loss will reduce, the scar will reduce, the lung function will improve. So this is one thing. The second thing is, along with time, you can also have real-time checking. That means once you finish the surgery, you can do a scan again to verify everything is normal and come out. And the third thing is surgeon's fatigue. Because you cannot concentrate on the same thing for hours together. You might have fatigue. So because you do it faster, the fatigue is less. So time, precision, fatigue, blood loss, all these are the good things with the robot. So that is the most important advantage. The other disadvantage is people with inexperience, okay, without good experience, if they start using this, because they don't know how to operate in normal, if there is a malfunction in the robot, if the robot is doing something wrong, because it's a machine, it can go wrong, you will not be able to correct it. So you will end up in severe trouble, and that cannot be corrected, and you need somebody to come there to rescue your patient, and all this can happen. That's the reason government is not everybody to use this technology. You need to be certified, you need to be experienced in this. Thank you.
How is robotic surgery validated before clinical use? How is robotic surgery validated before clinical use?
Validated before. See, once the robot is in place, there will be multiple bone models which represent the spine, 3D bone models. So people work on this technology for years together. So once there is 100% precision acquired on bone models, then they go to cadavers. So even before coming here, I had to train in a cadaver with a dead body. So we have to go to the lab, we have to use the robot, we have to operate on them multiple times, and then only after getting trained, we get a certificate. With that certificate, on the background of our experience only, we can go and operate on a normal chair. Before that, we will not be able to operate. So that is the way we will train ourselves before, and also the technology will be advanced before coming on to the real stage. Thank you very much, and thanks for giving this opportunity. There are many people who can operate with the robot, but there is nobody in India who can create a robot. I would expect somebody from here who will take up that challenge and come up with wonderful robot which people can operate.
Thank you, sir. Thank you for clarifying all the doubts from our students. And I appreciate all the students who actively participated in interacting with today's speakers. And now it is my request to welcome Dr. D. Prasanna Kumar sir, Samuel George Institute of Pharmaceutical Sciences, to kindly come forward for addressing the closing remarks of this session. Please join your hands and give a warm round of applause.
Thank you.
On the behalf of organizing committee, I would like to sincerely thank Dr. Sri Krishna Chaitanya Pondeti for delivering an insightful session, sir. Thank you very much, sir. Particularly the robotics in spine surgery. So today's session highlighted with the anatomy of the spine and disorders with the spine and how they treated the spine disorders, and he explained the different cases who age group people are suffering with the spine deformities. And sir, what you shown the videos, those videos enlightened the students and they thought that how the surgery was happened in the operation later. Thank you for showing the videos, and really the students got the goosebumps when you showed that the Halloween video. So the boy, when he started the walking, everyone applauded you. So for that, we understand how students actively involved in your session. And thank you for your enlightening knowledge here, sir. Thank you for that. And thank you to everybody, member of Culture Council, for giving me this opportunity to participating as a co-chairperson for this session. Thank you very much.
Thank you, sir. Thank you for your closing remarks of this session. And now, I request all the dignitaries, the advisors, the global secretary, dean of academics, co-speakers, principal, and all the faculty members to kindly come forward onto the dais to felicitate the speaker, chairperson, and co-chairperson. Give a warm round of applause on welcoming all the dignitaries.
Yeah.
It is our great privilege to welcome our speaker,
Thank you.
Dr. Sri Krishna Chaitanya sir. Give a warm round of applause for felicitating our speaker.
సార్ కొంచెం బ్యాక్ మీరు ఒకటి ఓకే
Congratulations, sir. I now request all the dignitaries to felicitate Dr. P. Kamreddy, Principal of Miller College of Pharmacy.
And now I request all the dignitaries to felicitate the co-chairperson, Dr. T. Prasanna Kumar, sir.
మేడం కొంచెం ఫ్రంట్కి వచ్చేసి ఓకే ఓకే మేడం
Thank you for all the dignitaries for gracing the dais.
What happened now?
ఏమేమి రాలే ఆ రోజు ఏమేమి రాలే
Ladies and gentlemen, as we draw the curtain on this enriching day to a total session, it is now time for a well-deserved lunch break. On behalf of the organizing committee, I extend my heartfelt gratitude to our esteemed speakers, respected guests, dedicated delegates, and enthusiastic participants for their invaluable contributions, insightful discussions, and active engagement throughout the session. Your presence and participation have greatly enriched this symposium, fostering an atmosphere of learning, collaboration, and academic excellence.
Lunch has been arranged for all the participants at the designated dining venue at Bronx of Diploma College. We warmly invite everyone to enjoy the meal and make the most of this opportunity to connect with fellow researchers, academicians, healthcare professionals, and scholars from diverse backgrounds. Let this break serve not only as a time for refreshment, but also a platform for meaningful conversations, networking, and the exchange of innovative ideas. We kindly request all the participants to reassemble at the Symposium Hall by 2 o'clock for the commencement of the afternoon scientific sessions. We look forward to another stimulating and productive session filled with knowledge and sharing and intellectual exploration. Thank you for your continued support and participation. We wish you a pleasant lunch and an enjoyable networking experience. The presentation sessions will be held after the lunch break in rooms 411 to 414, followed by rally. Participants are kindly requested to assemble in their respective presentation rooms following lunch. Wishing each and every presenter the very best, present with confidence, and may your hard work be rewarded. Thank you.
Order to disabilities globally, among 29 million children are educated. It is very important because the community not going to go to school and going to not actually contribute to society, and they have higher risk of common causes of mortality. In India, the census and all were not being discriminated in 2011. Census had shown a huge number of children with disabilities, yes, but they found 1.24% only. And that is something which seems to be carried out in study with in the Indian study, which led by Dr. M. Kurora and the scenario that was network coordinator of the project, in which we had assessed 3,964 children, and the sample was conducted by population proportionate by size sampling, so as to have a representative sample of the entire country. We had Kamram, Hili, we had Palma for rural, we had Hyderabad for urban, we had Ninkal district, Odisha for tribal, and coastal was rural. And we found overall 12% of children in the age group of 2 to 9 years had one or more developmental disorders, and 1/5 of them had more than one condition. So this was something important to be seen. Early child developmental index is a composite indicator to assess literacy, numeracy, learning formation, physical development, and social emotional development. It's currently used as a tool to measure early development of children 3 to 4 years old and children. And it has been estimated that 32.2% of children in India have a low early child development index. So the at-risk population, in the developing world, is estimated 50 million children, which is 43% child population, and increase is not achieving their cognitive development potential in the first 5 years of life. And they say at first 1,000 days, which includes time, pregnancy from conception onwards, are very, very important. And something which is important, and we have to understand, and you have, in India, you have 164.5 million children between 0 to 6 years. 46% have underweight, 38% have stunting, 19% have severe malnutrition. Stunting is a proxy indicator of suboptimal cognitive development, and you have micronutrient deficiency like anemia, which are also associated with poor developmental outcomes. So reduced mortality because of the increased care which is increased over the last couple of years. So sicker, smaller number of babies are surviving. So the reduction in mortality, you are having more comorbidity and developmental challenges in these newborn survivors. tied to OSC per se. It's a neurodevelopmental condition which is lifelong globally. Eventually estimates around 100. And we have to take off for how you see the climate and how the intervention related to the space and the health system. There have been rise in autism, not only because of increase of awareness, but in general rises beyond the classification. And there has been a rise of grade to 4% since 2000, and CDC 2025 estimates that the came out in January this year are 1.1. And our evidence study that showed how the data collected up to 2011, the population in the 80 was 189. Boys were 3.4 times better than girls. Showed that is like 40% of autism children had intellectual disability. This figures on the best what in my experience inspired and that in our population. So you understand that person spectrum disorder has issues of social interaction and communication. They have distinctive repetitive patterns of behaviors and interest. They exist in sameness, they have some issues. They have to keep their non-bound attention, their language, and the adaptive functioning, how much independent they are and how much support they are. And so there are whole lots of comorbidities like epilepsy, religion, sleep disturbances, irritability, GI disturbances, anxiety, genetic conditions, depression, motor problems, and etc. This is something important which I feel is important to understand. You have some genetic factors, you have the problem of religions, and you have a lot of variability. There is a risk of recurrence of 4% if a boy has autism in the family, and 7% of girl has autism in the family, and this increases to 1% and plus you also have 7 to 8% history of people having some behavior or personality issues in the family. So there are some genetic factors associated with some genetic conditions like Down syndrome, tuberculosis, neurofibromatosis. Then there are certain environmental factors, having viral infection, advanced father's age, maternal age, father's age, and mother's age. We did a study in which we found that there was increased effects in the scope of unfortunately fathers and children had autism, their fathers had regulation effects and sperm, and they could be correlated with the stress levels and the quality of life and lifestyle of our fathers. As in exposure of mothers, like pregnant mothers, air pollutants, pesticide exposure, and medications and all that. So all of these are there, and there are epigenetic factors that is gene expression is affected by the environment. So there are so much complexities which are there, which can come out of them. We have done a lot of studies on, we've done case studies basically on many elements and genetic association of many associations are the same. And in the study of metabolism, we found oxidative effects, we found non-glycation production. So there is vitamin D deficiency and lots of studies to understand, and there is a lot of complexity in this genetic environment and everything. So we can understand the complexity of the condition. What is the diagnosis? So according to this publication of March 2023, according to the Centers for Disease Control in the US, they found that the median age at the previous known diagnosis was 49 months. 36 months at that point in time is huge. And next time is important that those children with autism with intellectual disability having no leading age diagnosis, 1 months, and those who endowed and intellectual disability later, they get masked and they get missing. This 43 months who had ID, not have 50 months. So when the prevalence country provides the average age of diagnosis is continually about 4 years. It was actually for us also 4 years earlier when now people are coming to the 2023. We are also having parents coming from 1.5 years. We will have parents even the first year of life. Some children who started having repetitive number of movements in the father and mother and doctors and something was missing that they must be found. So other people are taking medication about the same. So actually the research suggests that we can identify them in the first 1 month at 6 months, the child is not responding to the gain communication is very normal, really, and it is very important. So we have to have a high index of frustration and there will be inconsistency in the aging patterns and phenomena. There are some children who show early onset, some of them develop appropriately after the performance and the energy. So in the guidance children do have a mind of times, but you need to have the button after the nervous systems going on, MRI studies, this, library studies, they can talk about skill that we have. Plasticity, now we can see great new genes and rehabilitation, and the early intervention period is the time that we know we can get so that when the child is most amenable to intervention, and I mentioned to you the first 1,000 days of the child's life from birth section has a massive breakthrough. So we need to be very careful. Why early intervention can change the development of the child is because their sensory periods are early increasing from the ongoing was high necessity for sensory performance. So you start the early intervention. It's important that genes are not destiny. Even if you have a lesion contributing to the experience of the necessity can be there as an attention earlier. So if you have something environmental changes, so that is important. And the intervention timing is starting in weeks to months after this, and then it was more rational than later. Why? It's important the intervention. Neuroplasticity is highest during the first 8 years of life. And the early intervention when it capitalizes this particular important because we show in trials randomized control trials that early social interaction model activity will show the significantly greater treatment gains than it was after 27 months. So, and other many other studies have shown the same. So, why it's important that in the first stage of life, there is a critically clear silent activation where surge of silent formation and the clean synaptic density and the blue one cortex happen after which there is silent the neurofunctional output will probably not should be optimized with early intervention as a lead possibility before the problems are taken as they come into the form. So then you can understand that there are outcomes in the intervention. The American Catholic Pediatric Research recommended to all children to be 1.5 years before. So it is something important, and this is true in autism spectrum disorder diagnosis. Today, people that are associated with a lot of considerable improvement in the social symptoms of these children. Now, how early is the early intervention, and what is this pre-symptomatic? We have to start identifying infants who have a very high likelihood of developing autism prior to symptomatic starts, so that maximizing the outcomes. So current research is going to support the identification of creative neurobiological model of autism as early as 6 months of age. So early intervention, we talk about preschool, years, and socialization intervention. People violation have demonstrated 25% improvement at 2 years of age a child's brain system and development. Pre-symptomatic is the intervention we talk about before 12 months of age, and target those with high likelihood of autism spectrum disorders, and not picking up the red flags. We are talking about red flags of big difference when you start early to be one. We are not labeling. Then there is something which is called pre-emptive intervention for infants less than 6 months, whether or not they are in shock cycle, neurodevelopmental. So, see, we are talking about very early. Something very important, primary prevention prevents suffering of a disease from happening. So if you know which are these factors, the risk factors are. We are trying to figure out autism only so that is by seeking treatment and we starting immediate management. And tertiary prevention is which comes later, and very supportive management where from the technological result and not technological insight. Primary prevention targets social and economic policies that helps address the government and society over the There are huge gaps in the organizing organization. their achievement gap is very, very limited. Such a very disparities in that resources they look for complex enough cost effective education system, the social culture gap. And we cannot just pick up in the high income countries, you can't pick up everything from there, they able to do some. And they are from the 5 million the largest and dealing. So it's very important and it's a national program trying to get any kind of special support for this language. This has been positive as well. So we have now 3 million, 5 million, evidence-based intervention and then it's scaling. So indications of screening for autism, delayed language skills, depression and social language skills. Anytime you have care, you have a concern for autism every time. And American Academy Pediatrics and CDC in Canada as well as Indian Academy Pediatrics recommend is special screening proactively for children at 18 and 24 months of age. So that is something very important today. They are talking about surveillance of high-risk children at each of the 6 from 3 years and routine screening of low-risk children at 9 to 24 months. And they have proactive for autism at 18 months. You do have the indicators less than 6 months if the vision is looking at risk, lab factors, decrease vision fixation. 6 months is reduced responsibility. Like this, you have by the 24 months, they may have no pretend realization of not going to be as useful to the communication taking place. There may be red flags of autism which we are all aware, and you see that sometimes parents say that yes, they are playing with children. You ask if they play with children or they are sitting among the children and playing on their own. So something very important, they do not play simple interactive games like back 8, 12 months, use few or no gestures by 12 months. Do not share interest with others 15 months. So we know all the whole list of red flags. Then there are a lot of screening tools, and other ones which we are using is a modified checklist for autism toddlers revised follow-up for less children, which is the most frequently used tool. We do have industry tools like this. 3 has been adapted. You can adaptation for 3 to 24 months. We have done social cultural adaptation for self-communities and the validation. And we also have things like the screening tool from 1 to 18 months of age. About in the those lines. For the diagnostic tools, yes, we do add-ons for research purposes only because it's difficult, or in complex cases, and because it's difficult to use in the sense because of the time takes. So, AIDS modified IMDT is a tool which we had revised from the Region 2 and very little based on dates and part of the guideline, and it's in simple access level. Then something important, and for which test which is most important for these children. The most important test, which of them. So there was a review which has been done, and this recommended that for the preschool, the toddlers, the use of combination of multidisciplinary assessment, including a pediatrician, a speech pathologist, and a psychologist, with other disciplines included depending on the identified abilities and needs, and DSM-5 or ICD-10 criteria. ICD-10 this paper was published that time, but now ICD-11 criteria in diagnosis, and to include information from clinical assessment and from childcare or educational settings, or as well as results from some instrument, especially for development of intellectual disability. And this is what we see that most of the improvements can be taken